Yeah, nice.
Refine it. You can come over and get two on. Back here.
Does that mean we're there?
Change the dive number. Well,
You didn't?
on the, I can change it on the fly.
Well, welcome everyone to today's dive. We are on board the research vessel Falkor, diving with a Sebastian ROV. And my name is Tim Shank. I'm a deep sea biologist from the Woods Hole Oceanographic Institution. And I am with...
Constance Sartor from University of Guam.
It's awesome. And we just reached the bottom at 3,376 meters. So that's, I guess that's close to 2 miles. Come on in here. Come on in here. There's Deb Smith. And what's this?
Hi everybody, this is Schmidt Ocean Institute, research vessel Falkor, and ROV Sebastian. And this is our lovely science team who just introduced themselves.
Anyway, so we
Come back here, please, Bridge. What was that?
Just sorting out our sideways movement. I know we haven't moved on the screen, but I'm just getting everything so we can get that trim on.
Do that.
So because we rarely have dived this deep on our 2 missions here that I've been on, we want to actually see if we can get a rock sample here so we can look at the iron manganese crust on it. We can look at the chemistry of the rock. We can look at the basalt, learn something about how the rock was formed, how this seamount was formed. This is a seamount that was mapped recently, right, Deb?
Recently. Yeah, yesterday.
Yeah, yesterday.
Brand
So,
new volcano.
right, brand new
No.
volcano. No.
Tim, these
Nope.
are all gonna be real hard to patch.
Okay.
I think maybe we just
Keep going up.
keep following up till we find like something real.
Absolutely. That's fine. It's just worth a shot here because we want to get one as deep as we can. So we have come down on a slope, and it was our intention that on these seamounts, they often have these limbs or these ridges that extend out from the central area of the seamount. And we are on the side of one of those limbs or ridges, and so we wanted to get this really a highly vertical slope to see if there might be coral sponges, see what's living here. We don't usually explore this deep for deep water corals, so it's a real treat for us. We're going to come up this slope. Not exactly sure if I could say how far it is, but it's our next waypoint to the top of the ridge, and then we'll turn right and go up toward the top of the seamount. See quite a bit of sediment drape here. I thought we'd see a lot more actually. This is steeper than I thought, but you can see on the right hand side there just drapes of our sediment on top of these rocks that look to be highly altered by manganese. So we will continue to look for a loose rock as we go upslope.
That's coral or spongivory.
Let's take a shot at it. Yeah. Top right.
That's right there.
Oh, there we go. I don't know who's got the ISO, if it's me or you. That's better. So we do have 2 sponges here. These are glass sponges. If I can get some focus here. Yep. One's got a short stem. One's got a
Yeah, I love it, actually. Favorite.
like a tulip sponge. I'll go further in. Let's see if it has anything living on it. All right, thank you.
And then there is the one right there in the nook, and then the one right there.
I got that one. Where's the... Not seeing much living on these sponges. Oftentimes they have ophiuroids or shrimp on them.
Yeah.
Oh,
Anyway,
let's keep making tracks.
There is also
Let me look up here a little bit.
something.
So the water temperature at this depth is 1.6 degrees Celsius, or close to 34 degrees Fahrenheit. So seriously cold, close to normally freezing temperatures. Of course, seawater freezes at minus 4 degrees. So no worries of this freezing just yet. What do you got? You got something? Or you're looking at that?
Yeah, it's coming up here. Let's not find it again.
Yep. So a little bit of raw exploration for today. Oh, there we go. Looks like a Bathypathes. Let's take a look at that, Chris.
Let me
Yep.
set up for it.
Yep. And Adam, is our ISO up high? It seems pretty little
yeah,
grainy to
it's,
me.
you can do your top.
Yeah.
Or you,
Well, I'm not.
try now, right?
I'm not. Yeah. I'm not getting much on it. I feel like it's on auto.
They're both off, but I can do it manually here.
I think that'd be great. So this might be a black coral called Bathypathes that we see at depths much higher than this. I'm gonna let you. So Chris is going to drive us into this. We got extenders on either side of the front of the vehicle that we'll just perch up and press against the rock face here, so it'll get nice and stable. That's how we're able to get really good video. There's always this inclination when you first see something, just grab the toggle here and zoom in, but that really doesn't help the pilot very much get stable and get in close. Just have to be patient while we sit here. What do you got?
Someone asked what
What?
the two levels are.
Don't have the oxygen levels up, but we can look at that in a second. Once Chris gets.
Yeah, you can. Yeah, I'm not even fine with that. So if you never went on that, that's fine.
You're good? Okay.
Is that a little better?
That is.
Now try your dial. Okay.
Yeah. For ISO?
ISO, yeah.
Yeah.
Yeah, there you go.
I think this is Randy trying to reference
Yeah,
something.
So it looks like a Bathypathes, and normally we'll see polychaete worms on this, we'll see crabs on this,
I know.
but no associates here, and that could be a function of depth. It's just too deep. But that's really nice.
I just lost that. So yeah, whenever you, I might have to go back a little bit actually, because that gave you no control.
Okay.
I can go back down. We're playing with our levels right now, so just
No worries? No?
take me away.
No. If you,
You got
if
control now, Nathan?
I do. I did. Okay. I got. That's me, right?
Yeah.
Okay, great. I think we want to get a little collection of this. Is that possible? It is. It's okay. And so we will collect things if we think they're new or we haven't really seen them before at a certain depth and whatnot. And certainly this is an observation we don't normally make at 3,368 meters for this black coral. So, all right, I think we're good. I think we're stable here. So I'm going to zoom in and get another beauty shot. You have lasers on for a second? Let's take a shot. See if I'm out far enough. Let's see. I'm all the way out. I am not the best to see these. There it is. There it is. Sorry. All right. Lasers are right on the coral now. Base. Okay. All right. I'm going to go in. We're going to take some shots, some frame grabs. Make sure there's. If I see an associate animal living on here, like a worm or a small crab, we will also try to collect that. So. see anything in here, do you? See anything?
No, I don't see anything.
I can't tell if I'm in focus either. Do you think it's in focus? The base is not in focus.
Three ticks.
Now that the base is in focus but the top's not, that's okay. There's a small holdfast for this black coral.
Yeah,
So I think, all right, let's get a little sample of this if we can. In a quiver.
Yep, watch out.
You got the camera?
No, you're good for now.
Well, I'll just hold it here. I'll hold it there till you need it. So, 2242. Depth is 3368.
Is this like a culture coral?
I forgot.
Bathypathes.
Bathypathes. Just got so excited about the wall that I forgot to
You have the whole thing, Kim?
No, just half of it maybe. Is that okay?
Alright,
Just half? Okay.
Yeah, I think so.
The side half?
It,
Top
it
half?
might be a little bit of a, a floater. So, more is better than little.
More is better than little?
Yeah. Maybe 2/3 of it would be good, just to make sure it doesn't float. You know.
A really good view here.
We're there, you're happy with that?
Yes, I am.
See how much you got?
Oh yeah, that's great. So our first number is 434. Excellent. That's in quiver 12.
Yeah. Quiver 12.
So when we get that back into the lab here, we'll look at the morphology. We'll look at the DNA. We'll look at the isotopes and a lot of other things that will help us identify what species that is and how it's living here, how it's related to other corals. So great sample. I guess I should have checked this.
Pretty RV.
Alright, Rich, you got it.
Yeah, we're gonna start going northwest, so I wouldn't bother trying to have a waypoint. We're just gonna move you from the ring line, so.
I think this is a big wall, so it's right.
Okay.
But I can keep one.
Well, I just didn't have to, we had that direction. Down.
I guess maybe we'll press this wall.
Shall I continue on this waypoint? It's no hassle to me.
Yes, please. No, let me just take a quick image of this thing up here, whatever that is. That may be a little coral. Just sit here another minute. Could just be a stalk of something that was once here.
Something above it as well.
Is there?
Yeah.
Just a slight bit.
Yeah, it's an old stalk of something with lots of hydroids on it. It's pretty old. Lots of fuzzy rocks. What, you had something else? There we go. There we go. Could be a little bit of a. Let's see what that is. I don't know if that was a bamboo or not. So I'm gonna get in focus here. Looks like this beginning of a bamboo whip. That's great. Okay. We can keep going, yeah. We've done a little whip up there too. Looks like it's the same thing. Okay, iris up a little bit. If you see something, holler. Oh, there's a little something here.
On the edge or something?
Yeah. You see that?
Yeah.
Get a little,
The starry thing.
yeah. We need to come in halfway and then drive it in a little bit.
Alright.
Let's come down a little bit. And do that. How many arms you see there? Oh, I thought that. Yeah. You got it. It's an anemone.
It's more like an anemone.
It's really anemone, but it's so low to the surface, you would think that it's waiting for any kind of thing to fall into its mouth, into its tentacles. So it's really low to the seafloor. Really interesting. Okay. Yep, let's keep going. Another stalk of something dead. Could have been a sponge stalk. Lots of sponges already seen quite a bit today. And our first 15 minutes on the bottom. Something, oh, is that a shrimp over there? Yeah, it's a shrimp on the rock there just above it. I'll go in like this. You can just fly it a little bit. See another stalk dead there. Looks like there's a lot of forams on the rocks. Not much current here, I don't think. It's been here. Okay. It's good, Chris. All right. So that's a sponge. Just looking about a corsanid shrimp, but it looks really healthy. It's got a really wide girth there. Maybe it has eggs. Still not. What's that? Again, don't see anything growing on that sponge of note. So I think we're okay there. All right, let's keep going up. Really wasn't sure if we would come in and start seeing corals right away or not. Oh, I think I've lost iris control. Here we go. So strange. Right, dead center, you mean?
Yeah.
Let's take a look at that. Looks. Yeah. It's a. Did you call it as a tunicate?
Tourniquet, yeah.
The gaping mouth. I'll let hold the camera there. Often has something living inside of it. Sometimes it's a copepod. I've seen shrimp in these things. They just have this mouth agape.
Those are just called.
All right.
They were all over the place.
Let's keep going here.
1 and 4 were actually completed. 6.
Cannot get this. There we go.
We got 2 cameras to work with that.
That's a coral coming up, right?
What?
That here, Chris?
Yeah.
Let's take a look at that. We might want to have to sit. That's a sponge.
Oh, sponge.
Yep.
So I've already done like filtering and filtered and smooth and it doesn't seem to balance out. So I don't know how else to further short.
Can we white balance on that? Or is that?
Yeah.
I feel like it's pretty white, but there's the.
Let's start.
There's another stalk over there. It could be a sea pin on the left. I don't know.
So I tried sending it to the
Oh, I didn't write it in here.
But then didn't do it the second time.
So, and probably.
Okay. We're going to set up on this sponge and just take a white balance here to make sure we have the right color temperatures for the camera.
If we came up, we were 3,500 meters.
We did both split up some. So I was able to I transferred the video.
We originally built it like 12:31 o'clock. And
Okay, we're just having a little bit of a watch change here in the control, mission control. So,
Actual position of the ship position. Okay, you're in charge of the log.
yeah, take them off.
It's good and good. Head on that flag, Jack.
Just a double change.
Hey, we're just really just getting going.
Yeah,
I'm assuming you've been watching.
I just caught from the black coral collection. Yeah.
That's mostly it, really.
Okay. So just
You saw the other little whip up there.
yeah, it looked like Candidella.
I thought it might be hard to get to.
Yeah, I mean it's tough to tell.
I couldn't tell.
I mean I put a message in Slack that has all the records of the corals.
I
and
just saw that.
Yeah.
Great. I'll go look at that.
Yeah. I'm not sure if Candidella was on there, but it could be an interesting
Yeah.
confirmation of that. But yeah, I mean I'm surprised we're seeing a lot of stuff
Yeah,
so far.
I know. I am too. The Panthers is right there. There's a. I don't know. That might have been a picnic on it.
Oh, there's something right there.
All right, here. You want to?
Do a white balance on that coral first. Thanks. Quite a few stops.
Yep.
Southeast of you.
Who's going to speak University?
All right.
So I'm doing typically 7:30 until midday, and midday till 1, and 1 till 8:30.
You need something? I've been chugging.
So I've just been relieving myself from breakfast and use this one. But otherwise,
Can we look at this stock here? This one? That actually could be interesting. I want to get a tight zoom on that. Since a lot of the stuff down here is really, really
you don't need to be up
fine,
8:30 at night.
we can't really work on snap zooms. Sometimes we have to sit down and get really close-ups of things. Do you have Slack on your?
I do.
Or you know, I've got it on my phone. What am I thinking? Okay. Yes. Yeah. Go ahead. Yeah.
Yeah, this would be pretty much a whole day, I think, for this side, so. You went to see it? You can leave it, like, just.
Morning. How are you guys doing? Sorry. Popped in here.
DVR roads were.
Can I just
Whereas in science or something. I do think, you know, well, they're like
Aloha everyone and welcome to the 12 p.m. shift to 4.
brain power to be just sitting there staring.
My name is Anamere
But I
Tenabe from the Brigham Young University of Hawaii, and I'm here on board with the scientist team. It's my very first time.
Hello, everyone. My name's Steve Oskovich. I'll be the other watch stander for this next 4 hours as we're diving the deeper parts of the US EEZ and the Howland and Baker, near the Howland and Baker unit, the Pacific Remote Islands Marine National Monument. But we're outside today. Reset, focus. I'm struggling here. All right. Very nice. So this is really interesting. So we got a primnoid coral here. This is almost certainly a genus we haven't seen on this expedition so far. The branching is really confusing me. Wow. What do we got here? A little amphipod? Try to get some focus on that guy. Maybe a little amphipod. Yeah, this is really interesting. Can we collect this and collect it at least here? You don't have to go all the way, but you know, somewhere around here.
Okay.
Under the second branch.
Do you have the
Yeah.
image you need?
We're gonna image it. Can you hit in situ? And I'm gonna do one more tight on the polyps, and then we'll do one more in situ.
Are you able to decide what the color is?
I don't know
Okay.
right now. It's a Primnoidae. All right. Can you snap a picture now?
Okay.
Very cool. Okay, we're good to go. So, if do you think this is big enough to fit in a quiver if you snap here?
Yeah.
Yeah. Great. Plus the branches, much appreciated. It is. It's a mystery.
Hello, Anna Mary and Steve. Hi, Anna Mary and Steve. I got a book before. Hi, guys. Thank you for joining us.
Can you grab a frame grab? Just hit the frame
Sure.
grab button while we're selecting.
No?
Yeah, just you can hit that anytime.
Okay.
Yeah. So this is FK21435.
Okay, so 1, 2, 3, 5.
Oh, you know what? I think we might have hit an important record on the CTD. I think we have exceeded the number of collections that we made in 2017.
Ooh.
Yeah. I think 420. 420s, I think, was the somewhere in the collection top.
And we haven't finished yet.
No.
There's more.
There's more. There's always more. Yeah.
That's cool. Time for enjoy diversity. No cancer, Good night, Diana.
So for this area, we are now diving in an area external to the Pacific Remote Islands Marine National Monument, but still within the United States exclusive economic zone. So while we're in the protected area, we were subject to certain collection constraints that are not in effect while we're outside the protected zone. So that's why we are able to make this collection. Very nice.
What quiver is that again?
This is going to be quiver 12 or 13.
I guess 13.
Let's see, 13 looks like. Yep.
13. Any additional text?
Primnoidae is the family. P-R-I-M. Yeah, I-D-A-E. That's right.
Family?
Yes. Yeah. Great. Amazing. Can you pass me another? Over there, there should be more of these things.
That will be the first sample from this ship?
Yeah.
Okay.
Ready?
Ready to go. There's another coral right there.
Oh yeah. It is. I think it's the exact
And
same
a sea
one.
star.
Yeah. Well, let's image the sea star. Since it's a new observation.
Oh, that's so quite like different a little.
Yeah. There. Okay.
Do I need to eat the?
Just a frame grab button is fine. Small polykey or something on the sea star. You see if there's a worm on
Yeah.
the arm tips. That's what I was looking for. This stick down here, I'm going to try and zoom in from here so we don't have to move. It's okay. Yeah, auto. Alright. Just a stick with a bunch of goo on it. Okay, we are all set. Onwards and upwards. Let's see. A lot of stalks here that are they could appear like corals, but they're mostly just maybe an old worm tube or an old crinoid stem. It's really difficult to tell which one is which. I think that's what we were looking at before. So a lot of the corals and sponges we'll see are going to be smaller typically at this depth. We saw some big sponges earlier before we came on watch, which was really interesting to note. But since the food quality is usually pretty poor down past a couple thousand meters, the diversity of life we see here is going to be very different from what we see or what we have seen in the past few days and weeks. Crinoid there. So how's it moving along the bottom here? Actually, sorry, one more. Yeah, I think that's what you saw. Right? Yeah. This is why it helps to have multiple pairs of eyes, because we're all trying to look for this really fine stuff down here. What we're not going to find these big. Did they end up collecting a rock? I don't have anything in the basket. I was just curious if it was written down. So we might do that. So this looks like a new observation for this dive of a different family of coral.
Also, the shrimp were white.
The shrimp?
Yeah, like their glass skins, unlike the ones like the orange one.
Yep. This is interesting also. Like, it's really interesting to be what is going on here? I'm gonna wait for some of the sediment to clear a little bit. Are we auto?
Just making sure everybody's awake.
This is a different kind as well.
Yeah, this is something else.
Whoa.
Polyp size is extraordinary here. One of the things that we find as we move deeper and deeper is that there's a propensity for corals especially to have larger and fewer polyps. And you can see actually the tentacles on this polyp are, if you take the tentacle span, it's more than double the size of the polyp itself, which is pretty extraordinary, and it's probably an advantage for them for capturing food particles and things. There is some evidence here for what looks like particulate matter capture. Wow, this is.
You want to go wide real quick?
Yeah. I can't even put this in a family. It's so bizarre. Sure. Yep.
Is this a sponge?
Um,
Down there.
That down there, we'll take a look at it. It might be a tunicate. I think this is a tunicate down there. That's really cool lighting, by the way.
Tourniquet?
Tunicate. Yeah.
Tourniquet.
Can I go in while we're?
Yeah, you're good to go now.
I think you're drifting. I'm pretty sure we're gonna set up for a collection here as well, but I can't even get a good firm handle on what this coral is, whether it's in one family or the other. All right. Can you hit in situ sample?
Sure.
And can you do any way to light the whole colony a little bit better? That looks okay. All right, we can hit in situ one more time. And let's go for collection, say maybe half. Half of it, yeah. Somewhere, around here. Yeah.
So if you look in the sit count, it's pretty big.
It is big. Really?
See the sit count?
Oh, yeah. Okay, whatever you can fit in a quiver.
You
So,
want to do about right where
can you? Do you have lasers on? Can you throw lasers on? We don't need to re-image it, but I want to get a size perspective.
okay. Pretty far away. Pretty far away from it. It's almost touching the torch.
Oh yeah, it's got like 3 dimensions to it.
Let me see if I can iris down my sit and see what's in it. So there you go, there's a prime example. You see where the lasers are on the sit?
Yep.
It's almost touching the torch.
All right. Yeah, whatever you can fit in a quiver. The problem is, like, with these deeper corals.
We can get multiples.
Yeah, no, just let's go for just one snip. Go for one snip, see what we can get in a quiver. And if it looks like we need more, we'll go for a second. Can you? Oh, yeah, that's all right. So for additional text, octocoral, OCTO coral.
Octocoral.
Yeah. Unbranched. I think it's a chrysogorgid, but can't be certain. Which is interesting. There's only one, from what I can remember, one unbranched chrysogorgid at this depth that's widely known. This will be interesting because it doesn't share any of the characteristics with that one unbranched chrysogorgid. Let's go for one more clip. Yeah, thank you. Can you write 2 branches in there? Two cuts. Yeah. Maybe half of that even. We don't need to take the whole thing. We'll leave some to regrow. And then before we leave, I'd like to image the polyps closed. Okay.
This is number 3, 4, 5.
Oh, there?
Yeah, right.
Yeah?
Yes. Yeah. So this should be 436, right?
4, 3, 6.
So the last one is 435. This should be 436. Yeah. and that is going in quiver 11. So what's the movement like down here? Is it like big layback or something for?
It's not too bad.
Yeah.
Not really apparent. It's real stagnant.
Yeah. Well, that's the real interesting thing is that these corals are occurring in an area where there is no current or very little current. That's actually really striking.
I think maybe they're just depending on stuff falling from a boat.
Yeah. Yeah. That looks interesting. That shimmering. Yeah, the Leonardo.
Manual now.
Okay. Yeah, wow. I don't even need to know. I don't even know where to go with that. Okay, can we snap a beauty shot?
Beauties.
Yeah.
Do I need to submit this one?
Yes, submit it.
There you go.
Wait a little. Reassure.
Okay, all set. All right, let's go. You know what? Can you test some of the rocks around here? Sorry, put away the arm. Can you test some of the rocks? Yeah. Just so we can say we got a rock here and we don't have to stop anymore.
Yes, sir.
Anything a little bit bigger in the 10 centimeter range? Yeah, yeah,
Right there.
Yeah.
Lasers are?
sure.
That one. Let's get you a quick shot of it.
Yep, that looks good. And if you could do kind of break it off if it looks like it's held
Okay.
crusty together. Yeah.
Give her a little poke first and see.
You got a little friend there. Can you hit? Well, just tap it first. See what you can do before we do imaging.
Yeah, it's loose.
No, we loosen the goose.
Not loose.
It's loose.
Okay. Can I do it first before you?
Go ahead.
All right. In situ image. All right. And go ahead for collection.
Okay. Slide away. You want to image it while it's in the...
No, just can you give it a little rotate, just so I can put some notes on it. Can you take a frame grab? Just press the frame grab button.
Now?
Yep.
Okay.
All right, turn it around. Just keep going. Okay, good. I got it. I got what I need from it. This is 437. So let's do storage.
Workbox.
The rock box. So where did that fall? Like back right corner?
Back right
Yeah,
corner.
back right corner. Okay.
Any additional text before I submit it?
Yeah. Rock, 12 centimeters. rusty color underside fell into back right corner.
Underside.
Underside, yeah.
Okay. Alright.
Back location in rock box's back right corner. Great. All right. Now we can move a bit.
Okay. Rock 12 centimeter rusty colors underside.
Well, we checked a few
location,
boxes there. Thanks for helping us through that. Now we can do some movement. So we dropped in kind of right on that blue point.
Yeah.
Is that
The halfway point
No.
between the waypoint and
yeah? All right.
yeah. Play with it a little bit.
So far, 1, 2. Oh, same thing, yeah. That's all right. We don't need to stop. So far, this site is very, very surprising. We've come across a Bathypathy species we have not observed yet, which was collected. We came across 2 other genera of corals, soft corals, octocorals we had not observed yet. So it's a total different transition from even, what are we at? Say 700 or 800 meters shallower. So this represents a pretty big step in terms of biodiversity turnover between the mid and lower bathyal depth range and the abyssal depth range. To date, I think our deepest dive was somewhere in the 2,800 meters. So, we've only moved about 500 meters deeper than that.
What is that? The sponge?
Yep, looks like a sponge. Probably, maybe not such a healthy looking sponge.
No.
Yeah,
So you've seen better days?
seen better days.
Is it still a glass sponge or a bamboo sponge?
It's a glass sponge, most likely.
Okay.
Yeah. Yeah, so this this is not a depth, as I mentioned, this is not a depth we've really explored too much. In fact, we haven't visited here, this depth range at all. Take a closer look at that.
Yep. Pretty stable, so you can just zoom
Yep.
in on it.
And here's something we haven't seen before on this expedition, at least I don't think so. Got a really nice collateralized sponge. Hey, Brian, if you're in the library over there, are these of interest to you, these collateralizids for sponge characterization? Just shoot me a Slack message or...
There's an associate on it.
Yeah, there's an associate on it, which is also interesting too. That's often the case with these collateralized sponges, so you might know these better as carnivorous sponges. They're very interesting, very diverse morphologies. But this is their home. They love these deeper abyssal depths. It's apparently advantageous for sponges. that like to feed on other organisms.
Get a good look at it.
Yeah.
This is a tourniquet.
Yep. Tunicate. Pretty steep here. That's where all the fun stuff is.
There you go.
Thanks. So we're going to image this sponge for hopeful identification as well as for associate relationships. There's a polychaete worm. That's the dark grain of rice-sized thing hanging out in the branches here.
So deep.
Maybe Brian was sleeping.
Yeah, I thought it was in the library. That's okay. We're just going to image it for now.
Trying to get some lighting for you.
Yeah,
sorry.
it's so well. It's not that. It's just that it's so difficult because it's going in 3 dimensions,
Yeah.
and so the focus is kind of wonky. I think we're okay though. Can you take a beauty shot and click
Sure.
that button?
Yeah.
All right, great. Let's keep moving up.
You got something up here to the right as well.
Yeah. Do a swim by.
See where?
Oh yeah.
Tunicate? What is it?
Where are you looking?
Yeah, big fine.
Okay.
Small.
No worries.
Just right here.
Okay. Wow. There is a ton of stuff here. I'm really surprised. So this is the same coral that we collected, just more branching. The first one. So it's a primnoid. But I was actually looking way back here at this cup coral, which is really amazing.
With that one? That's a bone?
Could be.
It's a deep one.
It's a deep one. That's right. Probably extremely fragile as well. I don't know what the depth range on cup corals is, but I know it's in the 3000s. The maximum observed. It's really low. Could we? I don't even know how to do a collection on that.
Section.
Yeah, you think it's small enough?
Might be.
I mean, it's pretty big. Yeah. You know what? Let's pass for now.
Okay.
Yeah, it's in a really weird spot.
It's a decent size.
Yeah, it's a good size. Okay, yeah, let's move on.
No regrets.
I know.
Ready?
So I don't want to rethink this, but if you had like a very low coral like that has maybe a centimeter off, how would you go about it? Would you try and like break it off with a jar? Would you suction it? I think it would be attached, right?
We used to, Australia, we were doing like we'll go on there with our jaws.
Yeah.
And
At the
get,
bottom?
you're at the bottom, just pop
Yeah.
it like this.
Yeah.
And then close slightly. And
Yeah.
Just balance it.
Yeah.
Bring it over.
That's what I thought.
And then, the ones that were difficult, the really, really, really small ones that
Yeah.
we're getting, those are all
Suction. Yeah. The thing I worry about. So were you diving 3 kilometer? Yeah. The thing I worry about is like suctioning something at this depth, it's gonna like really like obliterate it. I feel
It definitely could.
they're really thin, really fragile.
Yeah, the ones we were getting were those real long stone ones that
Yeah.
were growing on the side of like grains like this.
Right.
That was a little more squishy.
Yeah. So for our group, also, it's gonna be tough down here. But if we see any cup corals, especially multiple cup corals, like where there's two of the same near each other, we would want to get multiple because we have very diverse interests in scleractinians. Sponge. I think they imaged that one a bit earlier. I was at lunch imaging something that looked like that.
Is that a sea squirt?
Yep, yep. That's a predatory tunicate. Megalodicopia.
Seems like we have a lot of tunicate in this. Not a lot, but just like quite a common
Yeah,
thing in this area.
it is. It is. So this branch primnoid seems to be one of the more dominant. Can I do just a quick in and out on this little stalk? I think it's the same one. Density of the polyps looks a little bit higher. Just want to confirm. Manual. Alright, yes, it is the same. Okay, good to go. Yeah, that's fantastic. I think I mean, over the course of the dive today, we'll probably collect a few rocks, but I want to put some vertical distance between rock collections just to see if there's any sort of important stratification in crust composition, or that's another one of those mystery unbranched octocorals. Some distance between some of the rock types that we might find at this depth, and also crust composition, cross depth, start and get a better picture of the site.
That's an interesting sponge
Yeah.
right
Yeah.
there.
I think that's the live one of that one we were seeing before. You see how part of it looks dead, and it's also some hydroids on there. Just keep going. I really do love this terrain. I think I haven't taken a look. So I got off watch last night, and we were just touching down at the bottom before we were coming back up, and then I haven't actually seen the terrain in this area. But we're on the same track as earlier. We're just a bit shallower. Oh. Crinoid. Yeah, I'm very curious why. So we're seeing crinoids, right? We're seeing crinoids. We're seeing resinged sea stars. We're seeing shrimp in some cases, but none of them appear to be really interacting with the coral, which is really interesting. In fact, the corals have really no associations whatsoever. In a couple of instances, there were some small amphipods that we've seen so far, but really nothing concrete that we've seen multiple times.
So you can kind of relate that to depth?
I don't know. I think that's Tim can speak to that. I think.
I think it could be O2 levels.
Yeah, I mean, I don't think it's necessarily that. It's probably some sort of depth.
Is this another cup coral?
That's an anemone.
Anemone?
Yeah. So depth, there's a lot of interesting things that happen in the environment with changes in depth, and we use depth as kind of a proxy to to say, like, for pressure, basically. That's the only thing that depth corresponds to. But depth also has all these other covariables like temperature, dissolved oxygen, salinity, and it's really hard to disentangle the changes in those variables with the change in depth. So it's not easy. Just we say depth because depth is kind of just the easy way to get out of it.
Gotcha.
Yeah. It's hard. It's really hard to disentangle those variables from depth. But there's other variables too that don't necessarily correlate with depth. For example, nutrients in some cases, dissolved nutrients. It all depends on the water mass structure of the area. So, interestingly enough, here I think we're diving in a completely different water mass from the mid-bathyal depth range that we were diving in previously. So we're diving more in an abyssal water mass. Not to pull up my.
Okay, there's a
Oh,
coral
yeah.
right there.
Black coral. Yeah, we collected that one, a piece of it. Went on Tim's watch.
Something right here.
Oh yeah, there you go.
See it.
Yeah, that's
Right there.
exactly what.
Intertwined coral.
That is a really good eye. That's exactly why it's so difficult to study. Yep. Yep. It's so difficult to study corals here. We've seen a pretty good diversity here. It's just that it's so fine. Holy moly. Yeah. Yeah, that's great. So this is a chrysogorgia, or a chrysogorgid. Yep, I got it.
Try to get some water flowing and something a little bit.
Wow. Yeah, it's a chrysogorgia, so like a golden coral, you're calling them.
It's just like
Fantastic.
with a little bit different.
Yeah,
Oh, there's
no,
a
it is
little
different.
Anthropod.
Amphipods, yep. I'm also looking for associates in these
Other
colonies, because
associates.
remember how shallower we were seeing a lot of crabs and things?
Yeah.
But no associations here that I can tell, which could be telling. So we'll set up for a collection, I think. It's a bit of a bramble, so maybe just clipping one part of it, and I don't know. I can't even tell you where to cut, honestly. I don't even know where all the branches are. at this depth, we are. So, if you had to assign direction to a current, which way would you put it? Not even variable. Light and variable. So surprising.
Do you need a
Yeah.
picture for that?
Yeah, we'll take a picture. Sorry, I should have done that earlier.
That's all right, we're getting better.
Yeah, closer in.
Sample Institute.
Just let you know if you need lasers.
Yep. All right. Do an overall.
Okay.
You got it. Gonna pull out now to. Yeah. Can you throw on lasers? All right. Yeah, I can try and get it out of the shot too. All right. One more. All right, and then I'm gonna go tight in the polyps. All right, go ahead.
One more.
Yep. All right. So, where do we want to cut? I think maybe.
That last side, you just,
Is that what you were thinking?
yeah, that's fine.
Yeah, something like this. You know, somewhere here, so we get some good amount because there's not much tissue on these things.
Okay.
Yeah, so we need a good amount of linear branch. Yeah. Thank you. Yeah, as I was mentioning, we're in a totally different water mass here than we find in the mid-bathyal depth range. You know, around 2,500 or even 2,000 meters, and those differences in water mass. This water mass here is called circumpolar deep water in the equatorial Pacific, and I know it sounds weird because we're on the equator, but this water mass is actually has some connection with the Antarctic based on how the ocean currents move.
It does have an associate to it.
Yep, little amphipod. We'll see if he comes along.
Do you want a spot of these or do you want to go hand?
Can you do you think you can go like here and cut that, or is that
Come on for a second. Where are you looking at?
here and further down?
Okay. So I get both those pieces?
Yes.
Clearing up this little fork.
Right there is what you're talking about?
Yes. Perfect. Chryso.
Chrysal.
CHRYSO. GOR. Not Chrysober. GOOR. GIIDAE.
That's
This
it.
might be
Too big.
it might be a bio box.
All right, let's do it.
There's a little stuff we couldn't really see.
Yeah, no, it was so brambly. I honestly had no idea.
I mean, we could try to put it if you want.
Let's not struggle. Yeah, I don't want to stagnate too much.
What's the temperature here?
1.6 degrees
1.6 degrees.
C. Yeah, very, very cold.
Bio box.
Yep, in C. This is number. No, that's number's not right.
348.
438.
438.
No, great.
Down there. Nice grip, sir. Invisible coral.
Yeah.
Yeah, so just to give you, maybe the current is going
Tell me south to north.
a little bit like northwest in a way.
From the northwest.
It's going to the northwest from southeast.
Yes.
rough,
Yes, that's what I want to hear.
rough.
That's what I want to hear.
It's slow.
Okay. All right.
See how it's just kind of lingering
Yes.
this way slowly?
Yes. 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 Yes.
Like sidestepping in a way?
I love to hear that though. That kind of makes sense with what I was thought was going on down here. Okay, great. Thank you for that. That's fantastic. Alright, so what do we got? So waypoint 2, maybe 100 meters? Less than that, yeah. Let's book it. And
This looks like nothing but sand right now.
yeah, yeah, and
Small outcrop out there, but nothing too big.
I think the trek kicks off to the northeast and then we end up on a ridge. Well, that was fantastic. Bit of coral there. I'm really impressed in how much there is down here and how different it is. It would be interesting to see as we get shallower where the transition seems to occur. Yeah, feel free to acquire the hard bottom too, if you see it in the sonar.
There's a small little outcrops. This one here. See if this travels in. It looks like it. That's hard.
Yep, some sponges.
Carnoids.
Once we get an idea of what kind of what's the dominant or characteristic sponges for this site, we may want to look at sponge collections too, but I'm still trying to get a handle on which ones are more common. It seems like the large platy ones
Shrimp.
or bowl-shaped ones are more common. Crinoids are probably the single most abundant thing here. These Syrianthid anemones as well. These tube-dwelling anemones, Xenophyophores, of course, everywhere. These tunicates, yeah. Oh, yep. So I think that's similar to what we just collected. Yeah. Can we take a look at the associations with that?
I think there's something more.
On the coral?
Yeah.
Yeah. So the more I look at this, I wasn't sure at the beginning, but this one actually does look very much like Pleurogorgia, which is a chrysogorgid. Yep. Thank you. So we've got here? There's like a bunch of stuff going on here. Anemones. The polyps are massive on these things, which is kind of what we expect. Anemones mostly. Anemones. So this looks like Pleurogorgia, but from my identification, I think we're out of the depth range of Pleurogorgia, which is, depending on what this is, it might not actually be Pleurogorgia anymore. It was recently revised into a genus called Romulogorgia for the species Militaris. Let's go. So I'm not sure. I think we've got a collection. I think that one we just collected, that large bramble, is actually this. It was just this one's a little bit more well organized. And the branches are a little bit more clear.
It's got its life together. What you're saying?
There's a cup coral. I think that is, yeah. Yep. So lasers on. What do you think that is, size-wise?
Small.
You think you can slurp that?
Oh yeah, we probably slurp that. Let me sit up for it for a way.
Sure. I don't want to deny you your opportunity to slurp another cup coral.
Yeah, we might be able to grab that.
Okay. That would be awesome. It's much more manageable for us too, having the smaller cup corals. And we're almost at waypoint 2, so we're making tracks and we're collecting specimens. Best of both worlds.
Oh yeah. So he's like almost the size of the Nautilus.
So from what I could tell from the other one, it looks like it was like kind of like a pedestal, a very low pedestal. So it's probably attached, but like by something very, very low. Yeah, can you put it in situ, the button? All right, and then with lasers, one more time. All right, and go for collection. Thanks.
Okay, I'm gonna start with number 1.
Okay.
Pick up right in the middle of it.
So this is going to be sample 439.
Side, for sure.
Cup coral for the description. I'm not sure what it is right now. Oh, even better. It wasn't even very well attached.
Yeah, I'm gonna slow down the suctions and
There you go. Got it. All right. That went into jar 1.
index into 2.
So the jar 1 and submit.
Okay.
Yep. Thank you.
Watch out.
Mind of its own.
Yeah. Fortunate. Coming back up.
swimming sea cucumbers down here. Different species and genera than we've been seeing, I think.
Take some quick images of this.
Yep. This one will take off. I guess this fits the name for sea pig. I mean, it's not the classic sea pig.
Is that full bed?
Yeah.
You can't take off.
No, not without checking that ballast.
They eat sands.
Yep. Yeah, you can see the gut, right?
Mm.
It's full of all the sand.
Yeah. Sucking in the sands.
Yeah, so the part that's kind of above the head region is this big fan.
Mhm.
That's kind of what they use to swim, get up into the current. Wow, that is full of a lot of sand. Yeah.
It's definitely a sea pig.
Yeah. All right. Yeah, not actively feeding. Yeah. Sometimes if they sense the current, they'll take off if they need to travel.
They don't have 3 pounds of sand in them.
That makes it harder, but if you lose some of that ballast, it's easier to take off. So we're pretty much at waypoint 2. I'm not sure how. Oh, yep, little jelly. A lot of them down here.
Is there any jelly names on this?
There's no jelly.
Okay.
Yeah.
I just write it.
You can write jelly. Yeah.
There's a little sea star up there.
That's something I want to stop and look at, or?
No, it's the one we already sampled. It's just a bigger colony.
Am I driving?
Yeah. Thanks. Thanks for pitching it though. Trying to keep good notes here. Yeah. Maybe feeding. I wonder if these are invertivores or deposit feeders. This one it clearly seems to be in a perched position, suggesting feeding on something.
Probably sand.
Yep. I mean, there's a lot of organic material, right? A lot of this fluffy stuff on the seafloor is clusters and clumps of this sticky marine snow. So it could be nutritious. They could also be feeding on invertebrates or something in the sediment. Take a look at this stalk, since it finally has some associates with it.
Cool.
One fast moving sea star.
What time of his life you just had there? You're a man?
Got it. So we're looking here at a stereoschematid maybe on this sea pen. This is the first association we've seen between an octocoral and a brittle star. Some good imagery first. Any interest in Asteroschema on a sea pen?
Yes.
All right. So we'll go for a collection just below the sea pen. I mean, it's pretty tall, so we might need to do 2 cuts. I don't know. What do you think?
We're able to get that an associate from him up.
Yeah.
That should fit perfect.
Yeah. Let's do that. All right.
Did that sea star in the back get blown a little bit?
It was blown. Yeah.
Okay. I saw it trucking across. I didn't know
Is there
what.
one raptor on the base? Do you see?
A what?
Is there something on the base?
Something around the base?
Didn't see it.
An eighth of
I'll
conference,
take
maybe?
control.
Good.
Zoom back in. Yeah. Go auto. Worm tubes, maybe. I'm not convinced that's alive.
No. It just looked weird. That's
Yeah. Yeah.
Does.
Good catch. But yeah, we can do it in a quiver.
Good job, Nicola.
So let's get an image.
Oh, dirt.
Any way to clear up the
I'm not done with that.
Sample?
yeah, but when the dust clears a little bit, we'll do an in situ shot.
It's a sea pen with
It's a sea pen with ophiuroid, yeah.
How are we doing so far, guys?
We're at waypoint 2.
Yeah. Looks like it's been busy.
It has been. We have seen almost everything we've seen has been a new genus.
A new genus?
A new genus that we have not observed on this. Yeah.
Oh,
so it's all
okay.
different. It's all different.
Yeah, yeah. That we haven't seen yet, you mean?
So that
Haven't seen yet.
That Bathypathes was what?
Bathypathes like, yeah, I don't know. I mean,
How many of them do you think are new in general?
I can't tell you.
We haven't. Is this really our first dive in this depth in this region?
It's a totally new depth range. It's a totally new water mass. It's totally new. I mean, the rocks are just like very cohesive for the most part, and
Well,
solid. Yeah.
I guess.
Good.
Good. Check.
Good.
Exactly. Good job.
Ready to grab now?
Yes.
Happy Steve?
Yeah. Let's
Is
go.
it
Let's go.
happy me? Happy automatic.
Let's go grab. Hit the grabber.
frame grab.
Yeah. Just it's a snowstorm down here, so we'll just try and do what we can.
already did.
All right, and then we'll do overall, and then we'll do a snip snip. All right. Let's do the overall.
Okay,
Oh,
here.
can you focus it?
Frame grab too.
Daniel?
Yeah, 1 second. We're gonna do a focus quickly. All right, frame grab, in situ. All right.
And Sample Institute.
Yep, and now we can go for collection.
Randy,
Yeah.
I received an email from this girl. I don't know.
Look at that, one tentacle up,
Yes.
fishing.
That's coming up. We're gonna talk to Tara.
Okay.
That's Hannah.
When is this?
2:30.
Yeah, that's fine. Yeah.
Pretty cool, right? Maybe
Great.
even more.
Oh yeah, it might come out. I forgot about that. It might come out of the sediment. We don't need the whole thing if you can let the other half live. It might all come though. They're not really deep. It's the tissue is sometimes tougher to cut through than the
But
back.
the good thing is it's bent, so we can bend it into the quiver.
Okay, yeah, you can bend it in, sure. Yeah, it should be pretty flexy. So that sea pens are some of the most deceiving things, and oftentimes I've been on other platforms where they look so slipperable, right? But that that
keeps going.
rod that runs up the back of the colony is very rigid, or can be like a spring. And so oftentimes they get sucked up into the tube and then they kind of just spring out and then it's stuck between the ribbing. Is it corrugated? The suction tubing. Is it
Yes.
corrugated? Yeah.
Okay, this is number 439 or 440?
This is number 440.
Okay. 440. Biology.
So C-pen with brittle star should be in the description.
Quiver 10.
That's right. Yep. Like threading a needle. It does kind of look like funiculina, but...
What is this question? Other than perimeter defined solution?
Oh yeah, good question. So we have goals to sample certain biology on this expedition. Namely, we're most interested in the number one, the characteristic deep sea fauna of this area. So as we're doing these explorations and transects and surveys, we're identifying and collecting things that are dominant or could be rare for identification so that we can do our video analyses better. We're also interested in collecting organisms that are the target of deep sea predation or are being predated upon. Those have been some of our most common collections. We're also collecting bits of coral for respiration experiments to understand how they live and how they interact with microbes in their environment. And then, generally we're just interested in collecting things that have not been well documented in this area, so we can get a better idea of what is living in the deep sea environment here. So if you're just tuning in, we are diving a new site today, a new seamount, different from where we've been diving in the past few days in the Pacific Remote Islands Marine National Monument. We're still in U.S. waters at about a degree south of the equator. We fortunately, we would love to spend as much time in the monument trying to explore and document all the biodiversity we find there, but we have other priority areas that are of interest to our team here. So we moved out of the monument last night and we're diving at a site that's quite deep because of some interesting other data sets that are associated with the seamount. The Nautilus and RV Hercules dove here in 2019, but much shallower. And so we are doing a similar track up the similar side of the ridge that they had explored back then, but we're starting at a much deeper depth and trying to meet essentially where they started their transect. But we'll try and get as far as we can. plans maybe to do another dive at this site. And then we'll move on to a bit further south in the vicinity of Winslow Reef, but remaining in U.S. Waters. What do you see? The stick over there?
No, it's down below straight.
Oh, yeah, what is that? I thought that was just some sponge rubble. It's a massive barnacle. Can you put lasers on?
Yeah.
It's a massive barnacle. That's a 10 centimeter long barnacle. One barnacle.
They normally congregate together.
Yeah, I've never seen one that big before. That's
We saw some solid.
that's like this big.
Yeah.
Yeah.
Pretty big.
Wow. Yeah, haven't seen that before. Okay. Obviously not feeding, it's all tucked in. All right. Yeah, good catch on that. We'll be here at this site for probably the next, let me do my math. I haven't taken a look at the calculus of the dive plan yet. Oh yeah. Can we do another quick zoom just to confirm associate relationships? And you can get both of these, that's ideal. All right, going left first.
There's another question over there. Are any rock samples being collected on this expedition? Are there any geology related objectives for this expedition?
So we are collecting rocks. Our geological interests are largely in collaborative with some shoreside scientists through the U.S. Geological Survey. We are collecting rock samples for a couple of different types of analysis. A little bit tighter. There it is. Very inflated central discs on these. All right, that's one. Yeah, so far a lot of these this is kind of like backwards. We usually find more associations with the Gorgonians and not so much with the sea pens. But in this case, we've only seen associations with sea pens. And then we've seen amphipods associated with corals, but in terms of larger macrofauna and megafauna, just these brittle stars. All right, I think that's gonna be good. All right, let's keep going. They look to be similar enough to the ones that we had collected. Yeah, we can move forward then at whatever pace is comfortable for you all. Cover some ground. That's one of the things about working at the deeper depths is that, you know, sediment flows downhill, and so oftentimes in the absence of current, where it doesn't seem to be a lot in this area, you get massive sediment builds up and slopes.
Oh, no, so there's nothing. See if there's some outcrops.
if you were watching a little bit earlier when we were doing our collections, some of our collections, we tried to be as least invasive as possible. For example, if we can satisfy our scientific needs with just a snip or a branch of a coral, we'll take that and leave the remainder of it to grow, and they do grow back over time. Yep, another sea pen with oaf. Yeah, definitely these sea pens are pretty dominant here. Yeah, that one, does it have one? Yep, it does. So far, everyone, yeah. Not loving those corals, but loving those sea pens. I wonder if maybe the currents are better here.
Let's see.
Oh, not that I would expect them to be.
Not really much. Looks like it's coming. Yeah, it looks like this one's coming from the northeast.
Yep. Okay.
Which would make sense because we're going up a different ridge now.
Yeah. Right.
So it's kind of one's pushing up, one's going down. So it's all only slightly. Nothing too crazy.
Crop circles. Yeah. Some sort of fauna probably made that. A lot of sea pigs here. This is kind of their territory.
Sea pig.
Mm-hmm.
Stomp the ground.
Yep, wide open plains.
Back up. A little bit. Huh?
Syrianthid anemone, tube anemone.
Updated Whitman Bridge.
Interestingly, yeah. You know what's interesting? Even though we're much deeper, the life on the sedimented slopes and sedimented parts of the seamount have way more stuff on them than the sedimented parts of the shallower seamounts we were visiting. Kind of gives you an idea of some of the specialization that organisms at this depth are capable of. They're just capable of. They're specialized for capturing this kind of low quality organic material off the sediment surface.
Super.
Yep. I'll do a quick zoom on that one.
Okay. Looks like a little sea star implant in the sand right next to it.
Yeah. Can you hit the lasers? Cool. All right. So pretty big. There it is. Pretty big sea star. Pretty big xenophyophores too, about 10 centimeters. You got one laser. Oh, huh. Maybe one of the handles. Yeah. So each arm is maybe about 15 centimeters. So 30, 31, 32 centimeters across. Pretty nice. So that's a Zoroaster. No, I don't think there's any appetite for xenophyophore sampling on this one. Yeah, it's a good thing to note that a lot of these little microtopography of the seafloor in this area. You know what these look like? Are those beaked whale scours? No, you ever see those? The deep diving whales. Yeah. They have this like characteristic groove shape. Another sea pen with another stereoschema. Yeah, so the beaked whales, I don't know. I mean, we've seen them at crazy deep depths. I don't know how deep they dive, but I know it's on order of a couple thousand meters at least. Oh, can we look at the fish? Since they're pretty rare down here. Probably just keep it in autos when we're doing the fish. Yeah, I was trying but failing. It's kind of moving too. Yep. Yeah. For the fish, just keep it in auto, I think. And I think we'll also look at the sea pen. Oh no, it's a shadow. Never mind. I thought it was a darker sea pen next to the other sea pen. It was just a shadow. Okay. Let me do some math here. Yeah, flytrap anemone. Yep. And then tilt up just a touch. That looks right. I had to look up this one, but I think it's a type of ophidiid cusk eel called Basozetus. Double check on that name and ID, but oh, you know what? Actually, I might change my mind on that.
You need Tim?
Oh.
Hi, Rob.
It's got really interesting. I wonder if these projections are these feelers that it uses to search for invertebrates in the mud.
Where's your?
Cool. All right, coming out. Yeah, yeah, those look exactly like these beaked whale scours. We started, yeah. So there are some deep diving whales called beaked whales that have been known to dive to several thousand meters, and I'm just not sure what they do. There was a paper on them a couple years ago from some observations of these whale scours off of California. But I've seen them in pretty much every single ocean I've done exploration in. They're everywhere. It's not clear what they're doing. If I don't know if they're coming down and if they're digging into the bottom, or if they're using you're trying to basically scratch an itch or something. It's very enigmatic.
Still no big rock outcrops. Neither side.
Yeah, we'll move quickly then. As quickly as reasonable.
Maybe those I'll go 30, 40 meters, stop, and then rotate, see if there's anything there.
Sure.
Could do.
What's that?
Is it possible to, while we're cruising, is it possible to keep a little bit lower altitude?
Yeah, yeah.
Thanks.
I was just raising up to see if we saw anything.
Saw anything in the sonar? Yeah. Yeah.
So far, nothing.
It's on right now? Yeah, we always turn it on. Oh, yeah. Mike, maybe, on the way up. Since we do another one, you never know. Obama, the Gulf of California, no,
Hold up, same. Big Whale Beach.
Lots of xenophyophores. So there's so this button here, xenophyophores.
Which one?
So those are, I point one out. These things here, they look like small little blobs of sand,
Oh,
or sometimes they might look like this, more like brain-like. I mean, there's thousands and thousands of them here. Pretty much all of these things are little xenophyophores, single-celled foraminiferans. If you happen to see them in the frame, just tick that button too. Like these are xenophyophores. There's
okay.
probably more xenophyphore diversity here than
'Cause
animal
it's
diversity.
sandy.
Yeah, there's two kinds there. They like these sedimented slopes. One, they build their homes, for lack of a better word, out of these sediments. They glue them together essentially with mucus and stuff. Oh, monopsid isopod.
Here are animals as well.
Yeah, they're protists actually.
Okay.
Oh, can we zoom on the helisaur? On the fish? There are still sea pens here, they're just really small. Let me just go out until we stabilize. Put lasers back on? Very nice. Awesome. Looks like Aldrevandia. All right. Thank you for that.
Very drowning.
Yes, can we speak and move up a little bit? Roger that, thank you. Okay.
Is it possible to see the map?
Oh yeah. James, could you throw up the map where we are on the dive track?
I think it's also just a situation
Lots of this mucusy stuff. Bits of Phytotrias. Can we look at that thing? I think that's another sea cucumber. Well, just to check, just a very small one.
I think it's a sea pig as well.
Yep. Yep. It looks like. You can get a good idea of these xenophyophores as well. I'm gonna sneak in before the sandstorm comes. So these are the xenophyophores, and that's the sea pig next to it.
Okay.
All right, gonna come back out. Just wanted to get a good look at those xenophyophores too, since they're extremely abundant here.
The whole time since I went live. Said it's been like that since we went live. Nobody's complaining.
While we're cruising across the bottom here, I think this is a good point to point out that while we're looking for a lot of organisms that our science party is most interested in organisms that are typically associated with hard bottom habitats. There's quite a bit of diversity in the sea sediments as well, seafloor sediments. Yep, there you go. Just as I was gonna go into my diatribe about how mud is so diverse, come back into the rocky stuff. I guess they'll have to hear my mud is extremely diverse talk one more time when we hit the next mud patch. But yeah, so this is similar to what we were seeing down below, but these definitely more look like pillow basalts and things of that nature. Another one of those cup corals. Is it one of those cup corals?
Where?
Right there, to the right?
It looks like it, yeah.
Do you think that's also that same size range of slurpable?
Should be.
Just because we have a couple of folks who are interested in, yeah, I think that'll slurp up. What do you think?
Yeah. And
But-
then there's
There's
another thing. Never mind.
Let's go for one more of those because we have the one thing about cup corals is that it's very difficult to collect. Go into a different jar, I think, just to be safe.
Pick it or suck it?
Suck it.
Okay.
Yeah, it worked really well last time. They aren't really attached at all. They're kind of just sitting there.
Just relaxing.
Yep.
Maintain this out.
Yeah. So just gonna come into a position.
We will do a one image before we go in. So can I sneak in?
Yes, ma'am.
Alright, going in. Alright, let's do an image. Alright, and good to collect. This is going to be FK21-441.
RVG.
Cup coral.
Element number 2.
Jar 2. Yeah, this one might need a touch. It's got a bit of a pedestal. go. Perfect fit. There is a sponge in the background I'm curious about looking at afterwards.
Right there.
Chart 2 didn't come through.
Okay, chart 2, yeah.
What's that?
Chart 2.
Yeah, chart 2. This one is a little bit bigger than the last one.
Just a little bit. It's probably sideways.
The nice thing is that yeah, they're wide, so it has a big surface area.
We'll get the closest shape. There it goes.
Jar 2 still?
Yeah, just want to verify.
Awesome.
Pretty sure it's all good there.
There it is.
There it's on there.
Very nice.
Set up for 3 now. This is the first essentially keep going on. mine we were just under 14, like 24-hour days.
It's a tiny little sponge. We haven't seen it yet, so I wanted to get a good image of it. Just left. Kind of looks like a marshmallow floating on a stick.
This is our continuum.
Yep.
If this gets to be over 8 a.m., then this is still less than 8 a.m.
Is that
yeah,
like
waiting.
kind of like quite the same thing with the roses study?
It is. It looks similar, right? It's actually a sponge, a type of glass sponge. I've never seen one like this before. They're usually a bit larger.
This is like, if I ever see it that get past the 8, then
All right.
I think we'll see some of these hours shift around that may lend to more time period, which we want to.
I can see us, time to burn, and then do an extra hour, maybe free.
Is there any auto on it? Is auto on or off?
It's off now. You got
Okay.
manual.
Maybe reset the focus.
Yeah. Just gonna zoom out and zoom back in sometimes. Yeah, that's right.
There it is.
Come back in there. Sometimes it gets some monitors going.
I know it's small. It's moving around. Sponge just does not want to be imaged. There we go. Okay. Nice. Hold that for 5 seconds. And let's go. So yeah, we're the plan is just to head up slope. We were in pretty soft stuff until just now, a few minutes ago. So we're just gonna kind of crawl up, making pretty constant speed. I think we've seen. Sponges. Mostly sponges here. I mean, so these rocks here, you see how like it has that starburst shape pattern?
Yeah.
Those are pillow basalts that have just cracked apart, blown apart.
What happened?
The other ones died one.
That's an acorn worm right there.
Mm-hmm.
Pretty big one.
What is the purple thing?
An acorn worm?
To save it, save the seed, and then you start reopening it, it may not come back.
Can we go in a little bit tighter on that seed pen? We're just looking at comparing associate, the associates that are on them. So we're looking at the ophiuroid. have a collection of this one from a bit earlier in the dive.
Just clear the seed and see what it's gonna be for everyone.
All right. I think that'll be enough. It looks identical. Thanks. Acorn worms. Yeah. So far, we're not seeing any of the larger attached corals anymore. They're not present on this outcrop. And interestingly, I think it looks a little bit more crustier here too, and from my perspective. A lot more botryoidal textured rock surfaces. The tunicate there. Very nice. One we've seen before on some of our deeper dives. Thank you. Yeah, so we're not seeing, despite having this hard rocky crust substrate, we're not seeing any of those deeper octocorals other than the sea pens. They are very fine. A lot of the corals here are very thin, wispy, blend into low contrast backgrounds very easily. So helps having all of our eyes on the camera and screen to see what pops up.
So sure the tourniquet seem really enjoy this lift. Sea cucumber.
It's tough. Somebody's got to live here. If you can manage to live at these depths, you...
Can you snap in on this guy?
Yep, sure. Another Astera schema on a sea pen. Looks to be similar species. Can't quite tell, it's all balled up. Alright, great, thanks. Another sea star. We imaged that one earlier. Just noting it. Yeah, so we came up and over a bit of an outcrop. Now we're back in the sand and sediment. Very, very curious. Nothing attached on that outcrop. We saw a fair number of corals, primnoids mostly, but also possibly some chrysogorgids a bit deeper.
Another sea pen.
Yeah,
Seems like it's
mostly
just
sea
like sea
pens.
pens and
Yeah.
this area.
This will be a really interesting observation because by my accounting, when I was analyzing our 2017 cruise data set from PIPA, from the Phoenix Islands protected area, we didn't see any sea pens below about 1,700 meters. Granted, we never really went this deep. The deepest we went was about 2,500 or 2,600 meters at most, and we're seeing totally different fauna down here than we would see even at 2,500 meters, so 500 or 600 meters shallower. No tripod fishes yet.
Sea cucumber.
There was a tripod fish the other day, but I was not in the control room for that. It was neat finding though. They're not very common in the Pacific. Very, very much more in Atlantic species, at least in the Central Pacific. I'm not sure about the Western or Southern Pacific. the different species of sea cucumber.
Mm.
They all have similar color morphs, right? They're either translucent or darkish purple or pink. Quick in and out. Okay. All set. Shrimp. One of the swimming shrimps. Not seeing as many of those scour marks as we were seeing down deeper. There we go. Thanks for finding us some hard bottom.
I was getting pretty bored with the sound.
Let's take a look at some of those sponges. Yeah. Those are some of the larger sponges that we've seen so far. Ready whenever you are.
Sponge with a beak and
Oh yeah,
everything.
this is.
So funny.
Yeah, we've got a bunch of yeah, another one of those massive barnacles. Persingids, crinoids, ophiuroids, ophiocanthid. First ophiocanthid I've seen on this dive. I think. Yeah, it looks like an ophiocanthid.
You okay, or you good without us?
No, I'm good with auto. Yeah, we're just gonna kind of fly here like this. Very interesting. Sponge. There's probably a couple of different associated organisms inside the sponge even. They often have polychaete worms sometimes inside them. A glass sponge. Alright. Let's go back to my sponge guide. So if the track is taking us kind of off this, can we just follow the rocky
This is
bottom?
actually more over towards the trap line anyways.
Okay. Cool. It's so rare to see the hard bottom down here. I want to make sure we take advantage of every minute we can follow it. But it's interesting to know we might be seeing a transition kind of before our eyes. We're really losing a lot of the larger branching corals that are 3,000 meters plus, or even unbranched corals in that case. Most of them we've seen have been unbranched or very sparsely branched. The current seems to be a little stronger here. What's it doing, like northeast to southwest? Something like that? Yeah.
It looks like, yeah. Whatever that's that.
Come from east to north. North. East to west.
Yeah, I'm surprised. Usually we don't see as many fish at this depth. I've been meaning to look up that fish ID for a while. Just got too distracted with the sea pens for a little bit.
Is that the building here? What's that? Wonder if it's one of the buildings here.
Oh, I don't know. I was actually gonna look that up. It certainly looks like one of them.
It looks like one of the buildings here.
It could be, yeah.
I've seen some of those producers that maybe like they're not attached somewhere.
Yeah, can we, if you see one, can we take a closer look? The red blobby things.
Just saw two of them. No, they're flipping around.
Sea stars.
A sea star.
Sea star, sea cucumber.
Sea cucumber.
That little store around the corner?
Yeah, it might be a bracingid,
If you're
in which case they're both sea stars. I'd say more bracingid, but yeah, way more than 5 arms.
a little fish. Some fish.
At about 3,000 meters, I want to start looking for the next rock. If we get up that far, we'll see.
What is the?
Oh, big anemone.
Oh, it's green.
Yeah, it looks a little green. It's probably more brownish. Just the light being further off the bottom. Rock pile. it kind of gives you some perspective. You know, these rocks could have fallen down slope, but there's some larger slabs here that suggest that maybe there's some topography under the sediment here. Like, take a look at that stalk on the right hand side that's on the rock. Maybe on the rock. I'm getting vibes now. That's probably a sea pen again.
Isn't that the one that's controlled?
Yeah, they're everywhere. Can we come around to the side to look at it more on one plane? There is some interesting stuff here. Solitary hydroids. So we've got a primnoid with some zoanthids, and I'm going to try and get a closer look. Can we, yes, please. Can we poke this one? Yeah, poke it after I,
The ones that you're currently looking at?
yeah, just after I take a quick image here. So this looks like it's in the genus Narella, which would suggest, well, we'll poke it to confirm, but this might be the deepest known record of Narella.
You ready to poke it?
Yep. Ready for poke of science. Just get a few of the polyps to close. Oh, huh. That's interesting. Kind of went the other way on that. Can we collect a piece of this? Just a couple branches. This would suggest it's probably not Narella, it's probably Caltrophora something, but
Just 2 branches, you said?
yeah, something like yeah, here-ish.
You want to sample
Before the
institute?
fork?
Before the fork is fine, or after, whatever. Make fewer cuts is probably easier.
You want the sample institute?
Yes, yeah. You can hit the in situ button. Very, very long basal spines. So if this is Caltrophora, it's probably the deepest record of a species we may know about, or it's a species that may be totally new. Since I don't think there's a deeper record for this genus at 3,700 meters, but it might be a different species. Double check that. So this is FK 21-442.
Mm-hmm. Quiver 1,
Quiver 9.
Quiver 9. Additional text for that?
Yeah, this name here. CALYP.
That's Ursa.
Yeah, and two branches. Okay, and then before we zoom off, I'll do one more quick tight zoom on the colony. tight zoom on the colony before we take off.
Do you also have 14 as empty?
14 is empty. Oh, sorry. Yeah. Yep. All right, can you do one more? Just hit the, when it's in focus, just do the beauty shot button.
Sure. Now?
Turn out. There you go.
Manual now. There it is.
Okay.
Hold up. Let me go in a little bit more. All right, go ahead and do one more.
Okay.
All right, that looks good. I just want to do a quick scan through tighter zoom on these zoanthids and associates. A little sluggish. I'm wondering if it seems to work better if it's like incremental, like adjusting the focus as you zoom. You notice that?
If you focus as you zoom, it'll get all stuttered.
Yeah, okay.
Because you're sending the same data at the same time.
Uh-huh. Okay. Got it. Okay. Let's go. We are breaking all sorts of records today. Very interesting.
Like what?
Depth records, I think.
Oh, for
Yeah, we'll have to confirm when we get the specimens up on top. But we're extending down the deepest depth records for a number of possibly species, but definitely genera for the Central Pacific, which is kind of neat. A lot of the exploration that has come through here in the past has really not focused too much on this depth range for a variety of reasons. There has been, practical reasons in that it takes a long time to get down here. It takes 2.5 to 3 hours to get down to the depth range where we started at last night and earlier today. So it is a big time draw, but at the same time we find that when we go down here we find a lot of stuff that's totally different from what we're seeing even a few hundred meters shallower. And then on the other side of it, a lot of the explorations by the Okeanos Explorer and even the Falkor in 2017 as well as the Nautilus have largely been focused by community-driven goals of exploration in the upper couple thousand meters where you typically find a lot more biology and fishes. So this is a first. I was really, really surprised there's I'll have to show you under the microscope later, but there's the basal scales, so the scales on those, there's like these small calcium carbonate scales on those polyps have really long basal spines, which is only present in a couple of different species in the genus. Take a look. Should be neat.
Cool way to the right, isn't it?
Yep. Yeah. I'll take a quick look at that. I think it's a C-pen again, but there might be two ophiuroids on here, so this might be useful to document in a little bit more detail. Oh, maybe just the one.
C-pen.
Yep, still the same stereoschemated, still the same C-pen. So, all right, that's great. Thanks. James, where are we relative to our next waypoint? Throw up either the map or move over to that. Yeah. Let's go then.
This morning where one would be magenta and the other would be black and the same moving.
Yeah, so you gotta keep changing it back again.
Change it.
As soon as you click this one, you change the clock. Don't click that one, it stays.
I wonder if we just delete them both and make a new one.
Possibly.
That's weird. Hard to see.
Right away? Or keystone?
Could do a quick flyby. It's a persingid. Maybe?
Is an asteroid sea star?
I think so.
Or
Let's take a look.
kind of like the one that
Yeah.
you guys took as one of the sample.
Looks similar, right? No,
Mhm.
this is a persingid sea star type of asteroid star. We don't usually see them on the sediment like this. They're usually on rocky surfaces or sometimes corals.
Hello, here's Spirit Tuesday, finished with my calls.
It's in 5-4.
Yeah, that's absolutely.
It's a very, very large Xena 5-4.
Any problems?
I'm gonna zoom in quick before
It looks cute.
the
though.
snowstorm. Yeah, this is a really good example. It's about maybe a softball-sized blob of sediment and this single-celled foraminiferan. Very nice. At least 10 centimeters. All right.
guess he wasn't too far away from Rocky.
No, no, it's been actually really good.
Oh, but that sea star, like he wasn't too
Oh,
far.
yeah, yeah.
Because you say you normally swim on rocks.
Normally on rocks, yeah. Rocks or basically any structure. If you see them, they're usually kind of attached. They have these sticky.
That's a little
What is that? Oh, crinoid. Yeah.
thing.
Yeah, they're kind of stuck to the rock, with their arms out.
There's one of those, is that a crinoid? What?
Yep, crinoid. Yeah, feather star. The next sea pen we see, we may want to pop a Niskin.
Wait, what?
The next sea pen we see with the
Right
yeah.
here? That one right there?
Yeah,
Looks like there's one too. Just
yeah,
past there.
just in the vicinity of the sea pen. Okay, so this will be Niskin, whichever one. 5 liters, fine.
Taking one? Is that correct?
We have not taken any. Hit the in situ button. Oh, and then storage.
Water sample
Yep, this is sample 443.
443. What Niskin is it?
Niskin number 1. 5 liters. Fire.
5 liters.
Near sea pen. Yeah. 5 liters near sea pen. Yeah. Great. 443. Yeah. So we'll use that one for probably microbial analysis. Since we're largely we did a CTD cast last night on our transit down to, was it last night? Yeah, I think so.
Yeah.
Times relative. Yeah, that is a really interesting rock morphology. So we did a CTD cast last night, and we were sampling and filtering water through the evening, but we only do a CTD cast down to about 2,000 meters, so this is basically water that we would never normally have access to unless we did some, changed out a few instruments on the CTD unit itself.
Keep them on the
No, we've seen this sponge a lot. Can we do a little bit of sampling on this sponge?
Okay.
I'm very curious about it. It's probably one of the more dominant sponges. And I'm wondering if it looks pretty soft, and with these, we've had pretty good success of just slurping them up, kind of like doing a little bit of a shave. Or grab. I guess it doesn't really matter. We'll be interested in just like a clip sized, whatever's more efficient for you all.
I mean, I can flip it and put it in the suction jar, I can put it in the suction jar only, I can put it in the quiver.
Well, let's try to do suction because it'll save us some quiver space, I think, since we've
You
been
gonna clip it and put it in the suction, or just use suction?
just use the suction and kind of go up against it and
Well, here, we can show that.
yeah. I will image it though before you have a chance. All right, hit the in situ button.
Yeah. Yep.
All right, and then hold. Oh, sorry. You're still moving in? Okay. All right. Hit it one more time.
Yeah, so you know, Steve.
Okay. And then go ahead and hit it one more time.
Yep.
Okay. Coming out. And we are go for a sample.
Should be jar number 3. I turned the light on. Want shrimp?
Yeah, if you can get the shrimp too, that'd be
Full section?
since there's a shrimp associated with this sponge. I see it up on the top there.
You got full section?
That's right. There is another shrimp on top if you're
Oh, there is?
feeling brave.
Alright, full section off.
This is shrimp.
Yeah. Shrimp associate. So you see how fragile it is. If you, I mean, if it makes sense, yeah, that's fine. Break it. So this is FK 21-444.
Sponge with an associate.
If you can get that piece, that'll be more than enough.
I think that one's just gonna end up getting stuck.
Okay.
I can get another small piece. There's some in there.
Yeah, there is some in there. Yeah, if you can get another small fragment, that would be helpful. If you touch it with the blade of the hose, it will probably just shave right off.
I think you can't see it where the weight is.
Just make it easy, yeah. Thank you very much.
Exactly.
More?
You know what? I think. Can I see the jar? No, you know what?
You'll see how much falls there.
There's plenty in there. Plenty. Yeah, that's great. Thank you.
What cream jar?
Jar 3.
Shrimp's there too.
And the shrimp's in there too. Shrimp and sponge. Yep. Great. Jar 3.
basically need a hook to come straight up, so you just
Can we do one more Niskin bottle here too? Sorry. Just you don't need an in situ there. You could just hit that one. This will be Niskin 2. 445.
It's a water.
Water sample.
Okay. What additional text you want me to input?
Water sample near sponge collection, FK 21444.
Near sponge.
Near sponge, yeah.
Okay.
Yeah. You can even put the sample ID number for the sponge, FK 21-444.
Okay, 444
Right.
or 445.
It's near sample of the sponge 444. Yeah, that's right. Very nice. Ready to move along. Thank you. Yeah, there was a paper not long ago, from the eastern end of the Clarion-Clipperton zone, where they did some side scan. They could see these marks in side scan. They were really, really huge. The first time I ever saw these, we had no idea what they were, but it was in the Caribbean, and we would see these scars like this. They're all about the same length, maybe a meter or so. And depending on where you are, they fill with all sorts of like phytodetritus and coconuts and stuff, whatever falls into them.
How deep does it go?
How deep do
The waters get over the
Oh, pretty deep. Yeah, you could, yeah. It would. I thought, it might have been with a different ROV. I thought Falkor did a cruise at the Cayman Rise.
Yeah,
first
and that
one.
is probably either Nereus or Rokos, probably Nereus, I think.
Yeah, I think that sounds right. Yeah, that was a long time ago.
Yeah. Probably 2014, 15, something
Yeah.
like that.
I was out there in 2013. It was a lot of activity in that area.
When whales do this, is they simply why they do this?
It's all speculation.
Scratch and itch.
Yeah, scratch and itch. Maybe they're looking for something, but I would think if they were feeding, they wouldn't be all uniform like this, right? They'd be kind of just like, I don't know, more foraging and less kind of uniformly arc shaped and of a known depth. It's fairly similar depth. don't know. Yeah, I wish, you know, I've never even seen a beaked whale, honestly. So I couldn't even tell you what these things look like other than from what I've seen on the internet. But, you know, it's totally possible that these things could be hundreds and hundreds or thousands of years old because of just how slow sedimentation is in this part of the ocean. You know, if you disturb the sediment, for example, say Sebastian was to sit down and leave a footprint in the mud, you could probably come back 5, 10, 15, 20 years from now and probably see that exact same footprint, almost exactly the way it was when we stepped down in it. So it's a pretty slow sedimentation rate.
Not a lot, but it's something.
Well, it's very
I mean, it's really interesting. I wonder how cohesive, like, if this stuff is stuck to the bottom, if there's more that we're seeing that's just buried by sediment.
Oh, look at that.
Tunicates, yeah. Flytrap anemone. Yellow bathy crinid, stalked crinoid.
Yeah.
Wow, yeah. Happy to move along, yeah. Yeah, somewhere around, so we got down around 3,300, I think, on the bottom depth. 3,357 meters. Maybe we'll come up to about 3,000, another 100 meters or so, or 75 to 100 meters, and we'll look for a rock or something like that. Try and stratify our rock sampling a little bit.
That's a real big patch right there.
I know. I'm optimistic. We've in the past, even 100 meters of vertical, we've seen some pretty good rocky patches. So
Should have just grabbed one there and just held it, just in case we get up there and it's just sand.
just plant it. Oh yeah, that'll go over real well. Is there a way to cruise a little bit closer to the bottom?
Yeah.
What's that movie on us? Shakedown.
They're doing some shakedown ops start of the season. They have an ROV cruise coming up July, so they're just doing some testing. Yeah, tripod fish. Good. Somebody was mentioning that earlier in the chat.
Tripod.
Tripod fish. Yeah, sure. Not too much depth of focus here. You should be able to. Beautiful. Now there are two genera, and I can't tell them far. I used to be able to, so I'll have to look this up for you all. But get some. Kind of. Well, might be 2 or 3 genera, but it varies based on fin structure, but also their eye spot orientation. Do you have an oxygen value here? Just curious.
Is 163.7 microbes.
Cool. Yeah. pretty well oxygenated by oceanographic standards for biology.
Is that a seapan as well? Down there.
Those look like worm tubes, and I was I might want to ask to look at the next one since there were some small projections that I thought were hydroids, but we should double check to confirm on the next. Yeah, yeah. Either, well, I was thinking like this one to the right of the laser is 3 o'clock. Those white stalks. Yeah. Can I just click in and out on this one to see if it has these hydroids or what's on this? Yeah, this is a type of worm tube. No hydroids. Looks like there's a worm in there though. Okay, thanks. Actually, there's another tripod fish, different genus, actually, right here.
Well, that's one of those fish without the eyes.
Yeah, they have, they're just eye spots. Wow,
Yeah,
this
they're
is,
big eye spots.
yeah. Two different tripod fish in as many meters.
Yeah, they were just talking about this one.
Uh-huh. There you go. So this, I think this one is Hypnops, if I remember correctly. No, Bathythylops. All right, I'll trust you on this one this time, so I can pull up the guide to check. Good.
So that's a tripod fish?
Yep.
But it's not standing up on its tripod?
A little bit.
All right.
You push it through the
That is true.
Ready. Yeah, I thought that's what it was, Hypnops. Hypnops is the one with the big yellow eye spots. I think there's a bit of a delay from the comments that we're seeing, so that's why I'm trying to read your comments as they're coming in. Depth of our next waypoint should be somewhere in the, let's see this, 2888 range. Oh, can we look at that jelly? Sorry, just passed it.
Where's there?
Behind you, just a meter or half a meter. And there was a jelly there. Came out of nowhere.
Was it swimming or?
Yeah, it was swimming.
Yeah.
There it is.
Where?
It's
Is it?
right there. Somebody was saying earlier, was it one of you two maybe? Yeah, that you saw those tiny red jellies.
Yeah.
That's all right. Oh, there it is.
I got your manual focus.
All right.
I was not going
It's drifting out of focus faster than I can adjust. Alright, I think that will leave it though. Well, you know, 3,000 might be a good depth to start finding a rock, because we're going to be in the middle of that slope. I think it sounds pretty good. Some cucumbers. Fan favorite.
What's that? That was the name.
Alright, so we're moving a little bit more quickly across the sediment until we require some of the hard bottom. Maybe there's something up,
Yeah.
something, a little bit of something out there.
If you
What are
see
those
any,
intervals at?
What's that?
What are those intervals?
30.
Yeah.
watery or any wispy sponge part, please collect them for me.
It only goes to 50 and then it won't be way off. I think it's just 70.
Is that a big rock?
Starting to get into it, yeah. Coming up rather fast too, about 30 or 40 meters just in the past few minutes. It's really surprising when you're moving over sand, you don't realize how fast you're coming up sometimes, since you don't see the terrain. Some more crinoids. Stalked crinoids. More stalked crinoids. Yeah, I think we've transitioned. I mean, it's pretty conclusive at this point. We've lost almost all of the corals that we saw when we first set down in the 3,300 meter depth range. Very interesting to dig into that a little bit deeper to figure out. Oh, now we're seeing umbellula. The umbellula sea pen, which is more characteristic of the depths we were at the other day. The that sea pen we sent. Oh, remember that?
Yeah.
That looked like a rosebud.
Umbrella. Oh, there's a fish.
Yep. The Basazetus. I don't know.
Whoa, whoa. It's quite big. Is this a catfish?
It's a type of ophidiid. It's called a cuskio, I think.
Did you see the sea star over there? Is it a sea star or anemone?
Looks like I made it.
Oh, I think that might be a crinoid or persingid. Actually, that might be interesting to investigate.
Yeah.
Do you think you can make your way over here to this sea star?
Sunstar?
Yeah, some kind of star. I don't know.
Whoa.
Any ideas? I've never seen a star like that at this depth. I mean, it's
So big.
probably something deep sea sunstar, but we'll image this pretty well since it's large. There's a crab on the rock. Didn't you see that squat lobster?
Is it?
Right above the star.
On the next shore cap? On the left?
Yeah, I did see that. I was going to ask you about that maybe afterwards. But there's a squat lobster here. I was wondering if Tim had any interest.
Should I get to Chris?
Yeah.
What's that?
I'll try that, Steve.
Okay.
What do you have for storm chambers left?
We've only taken 3, so we have, including the flush, or excluding the flush jar, what, 6? No, 5.
Frame crab?
Yeah, can you frame grab? Just regular frame grab.
Okay.
What's the top of that waypoint?
2888.
I think I've seen that one.
2880.
It's very, very small.
What kind? What do you?
Yeah.
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 Okay.
I think I really need some of these for background too.
Okay.
Be great.
We're gonna do some slurpage.
Badges.
Want some sample institute picture?
Yeah. Let's take it in situ photo.
It's like a weird.
Okay.
Okay.
Wow.
Coming out, and then we can start the collection whenever you guys are ready. This is going to be jar number 4.
Four,
6.
6.
Yeah.
Squat lobster.
Squat lobster.
I realize it's gonna be for.
with deep heavy filaments.
It's what?
There's a question on there.
Oh, I don't know. No, I didn't see them. I think they're talking about the fish, though.
Oh, okay. You want me to take it?
Yeah, in like a,
No.
when it starts going in. Yeah,
Okay.
that's good.
Good. I didn't have to ask you.
No.
I mean,
You thought
yeah,
you
I
would?
mean, so there are two known from the Central Pacific. There are two known bamboos.
Yeah.
Both are Bathygorgia.
Yeah.
And I'd be curious because Bathygorgia doesn't have any evidence of branching. Was from the best of my knowledge. It'd be really cool to see if this is Bathygorgia. It might be actually.
Well,
and
does it?
might be
Bathygorgia,
branching.
Bathygorgia has polyps all on one side, right?
Yeah, yeah. This
These
I
are
can't
sort of
tell.
on
It's
side.
kind of right.
Yeah.
Because there's a northern hemisphere Bathygorgia species and a southern hemisphere
Mhm.
Bathygorgia species. So it could
And
be
another one in the middle.
maybe.
Man. Well done. So that's going in quiver.
But we can key it out using the brand new
Yes.
table.
What quiver is that?
That should be
You're 14.
14. Okay. A French bamboo, yeah.
I think we're making really good time.
I think, yeah.
On dive track, I think we might make it up past
Cool.
2,500. Yeah.
Yeah. That's great.
If everything keeps going to plan.
Right. Well, we're getting some good quality observations.
Yeah. No, it's definitely. Yeah, all of the same Mystera schema on the sea pen.
Yeah.
Didn't see any variation.
No. I think they're- have you
But
seen-
it is-
have you seen crabs on anything?
No crabs. This is the first one.
I didn't think so either. Right. Okay. That's a good sample right there. Hold fast too.
Yeah.
It's really nice.
All right.
Automatic. I don't know if you know this, but I have a Facebook page. They posted that you are broadcasting live right now. A lot of people can
No,
hear this
no.
on space, so you should definitely
Keep
take
going
some
up another
video.
30 meters. If it looks pretty clean in terms of rock collections, in the next 30 meters we'll stop short of 3,000. But I'm hoping it stays just like this because this is good stuff.
So which place are we now? What place are we now?
We are in the Seamount, an unnamed seamount on the Tokelau Ridge
Unnamed Seamount.
in the U.S. EEZ.
Okay. Okay, for those who are joining us from Kiribati, Maori, and Ngai Sera Racemen and the Seamount, the unknown seamounts in the U.S. waters, ibun wagin ang kay tiran ari the life ibugin the dive, hopefully ba kam China sa kam gunan na nga amisi sira kay kamay sin den tiran ari ini comments bas anagaya kagi kamra ba. O ye dari kirang kay Dr. Randy o Steve nga ye danan scientist agi amin ang detaya yo o Tim ako na ni Bubo ibugin na misi sira nga naro na pa ye misi sira ibugin the dive ayo.
another one? Is that the one under the rock? Can we zoom in on under the rock?
Where did you see it?
This one?
Oh, yeah.
Is that the spot lobster you're talking about? No, two Bersingids. Oh, okay. Yeah, I think that's the one we sampled, but we can do a quick flyby. Yeah, it is interesting. Like everything we saw at 3,300 meters is now disappeared and replaced by all of these. I don't know what we'll call it. 3,000 meter assemblage.
So it's branched bamboo. What is it?
Yeah, very sparse.
Maori nakatitigi. Ang Canada ko may kanere sa simerang kandiran na Indian known Seamount in the US water kanaro na pa iya misi sila kitirana kam gona ni Korea. Ikan niya ako na ni na kay Dr. Randy o Steve o team da ng scientists ay ka-join us in the dayayo in the ship tay. Kana yay na kain to tanan lady kaya kan nora kitirana anang guguray na kain ni Busmaya. Kay sa ronde tapari ikiribapa kaya nagmais tanan na sibuyay. We hardly seen anything from in this area, but hopefully as we go up, we will see more.
Alright, I think we're gonna start to look for a rock, but I want a very specific kind of rock that kind of has this texture on it, the bumpiness. Something over here.
So right now we are looking for
There's
another
like-
collection
there's
on
some
a
at the
rock.
very bottom.
So they're trying to figure it out which one is the best one for the sample.
There's? No. I'm not sure. Yeah, we can try it. I mean, this one, right? This one. This one. This one. This one. All right. Yeah, if you can break that. Yeah. You see how there's like a little cauliflower shaped.
And for those who are just joining us, just for your information, we already have been collected a lot of samples already. So if you do have any question, feel free to write your question on the group chat or the live, and then they can able to help you to answer your questions.
Go ahead and try and break it first, and we'll image it later.
Yeah, I'm just trying to decide which one I want to use.
It's good size, like 12 centimeters, 15 centimeters. It's a good size.
Yep. Mhm. No worries. So, aneng kanit ya sibo na nakibugin di sampo na harawak.
That's a great one. It has a bonus crinoid on it, so good eye. I know that's what you're trying to get.
So usually nakaya interested nira na rin the coral men dita yayo tamen danan colleagues sa tangira ti sample yon the rocks ngayon na ani bugina daw na yakapungan na yata yay ni collect sini danan asib
If you can just put it center screen.
ibugia
I'll grab
tamen
the fish, yeah. That'd be
ibugia
great.
ya colleagues tamen.
I've got plenty of.
Kanga in general sa si the research sa the expedition na yosi bugin the coral me ibugin bayay ka mais o
Yes.
tanan scientist ay kayo kakokoro ya interest yon
Stow it.
paintari ngayon ya kadayan na rin ni make it fair na ko yan ni kapeni ni collect sa ke bayay ki ako na ni collect si bugiya tapi men na ba.
Thank you. Excellent collection.
Oh, I'm so
No, I
sorry.
got it. I took it.
Kay si Vera ki bugin nam rong otte katiti ki si Vera ki sa giruri ka access pa maing may na no make up in may yantari ki kanga ro yaro na may ina no rin ang takabenyan tip ang kay sa tabi ni Vera ki nakweta. Ang kay sa bariko na narita na nasibu. Meuri lili sa bariko ita na nasibu. Kaya na kay kanga anang kara kumata na ni koron ndigin tetap wayo. So nakaya ya ayara na re ya interest kanga bunyon tira na rite coral at men de sponge may bugina pa ya colleagues tab men na ke atangiri ni kan kare ke ine danan na rite nan sample yon danan rocks kita biya wari ki pain tari ngaya ya I make it fair na ni ko ni ka bunga ya ta yo ice but the rock nga yasan na ko to kami sa ke may na may spaya kya san ni collect na tana ni coral me tana na ri tana na associate ke me na yana
Yeah, we can do a quick snap.
how deep is the
Right now?
this tide?
We are 3,000 meters.
Okay.
Yeah. We started at 3,300.
So si start maintenance with 3 tinwat nga tinibubwa ang kay saro ko kanga saro kon 300 meters sa tabi ni Kekerak.
Oh, spot lobster also. We'll image the spot lobster first. On the left side. Just in case it swims away. Yeah. I just don't want it to swim away. So it was on the move.
Tin sa buare iga ari kana na yeri pa ya kia ari ko rikuwa pa ngaya na karawag ta kami te pa yana. So are you guys trying if you collect the sample? Yang yang siram si sira kani. Te teran ari akaten di semen ba kanga irawa yari di kitanan te koro me teran ari tanan rock ki the substrate age to ako na yen ran ari determined by ay menra to pa ay kay ni menay tari. Ang ay based on the age.
Oh, another one with eggs. Looks like immune to spot lobster. Okay, I'm gonna pull right over to the other side and the coral get back quickly. So we got another Pleurogorgia with barnacles and solitary crinoid. Again with this zoanthic, it's kind of like the same one we saw a while back. Alright, good to confirm. Alright, ready to go. Thank you.
So ang kay paya kisi aki ari gori ko aki kanga paya ki abo o to punta February ki paya ki apun sa nori may maina nakakinda na ni koro tiyan tanan na sibo mamay siri gira ari pun pain tari ki ataku pa anang kakawa ki pa na ni collect sana ki bugin karawagin ta kamete pa yon o ta kamete pa ibo puto yon to kakaya na pa irawa ayariri ki paya kay ka ayrawa ang kay tiriri ki asan kami na to itari ki kanga i-base on deriri ki. Let me know nga na yansa mi question ke yagi nang para roy. I'm sorry.
No, it's great.
Trying to answer the question. So.
Snail. See the snail?
Yeah.
It moves by kind of doing this
Is,
rotating.
is that actually the first snail that we've been so
It's
far?
the biggest snail we've seen. Maybe the first snail, yeah.
Like, I mean, in this type?
This dive, yeah. There's a lot of Pleurogorgia in this area. Interestingly, I'd expect to see more too, because the Pleurogorgia is one of these corals that loves these vertical faces, and they can get very, very dense.
We hardly seen like other kinds of corals, except that on the sea pen
With sea
and the
pens, yeah.
primnoid.
Primnoid, and we've seen a lot of these small cup corals too, right?
And cup corals, so true.
Yeah, this is kind of classic Pleurogorgia here.
Is that looks the same, the similar one with the one that we just took?
I think
Lately.
yeah, it's almost exact. Although you can see actually this one is really nice. It has the polyps closed.
Mm-hmm.
There are a couple species of Pleurogorgia. They've been recently revised, but this one has the polyps closed, which helps us also get a better ID in some cases. But we do have a specimen, so we're just gonna get an imagery of polyps closed for video analysis, and then we can move on. Just hold it for a second. And that's it.
So, the wet ang kay kangay ng yarari yan ang inang may turo sa ro ang kay kangat timina 1.6 Celsius. Ngayon, using that on rocks ay kay.
so by growing very slowly, we can, we have some assumptions that they're very old, but in many of the species, we don't know exactly how old they are. So we can use some information that we collect from these corals to identify,
Determine.
how might, approximately how old they could be, using a couple different types of analysis, and also what they're feeding on.
Ngayon nakatitige wasigo Denan Rock. Okay, ngayon nasang. I think pretty much the same question that I asked you. So she wrote it again, like if you can know like from the weather. Wait.
Like, how they've been exposed.
I think so.
I mean, we know, well, looking at the rocks, we can tell a couple things. We can tell, what kind of seafloor we're looking at. Is it something that's made from reef, like from corals in the shallow waters, or if it's volcanic? And what we're looking at here is pretty clearly volcanic, but there's a very thick layer of crust on it. But this is largely volcanic, came from a volcano at some point in history. Now, take a closer look at that. Okay.
Hopefully behaga si sirak nakatitige.
Okay. Scratch.
Men nakabara-para to wayo ito ko pa kukunan ranari. Kukunan tao pa tira te wete ang kayay ito kukunan men nuragin bai ki yon tira nari tia sibu kita nan wa kay. Men am observation na kikunuri men te wa kukunan tao pa tira te wete. Baya kakira tin te weta.
Yeah, well, no, just go on and poke and zoom in on your finger as you're touching it.
Soft poke, hard poke.
Soft poke first.
Tickle, if you will.
It's like a,
So ako na nadaya pa kakira tin te weta men taragin tira nari men te wa ka kianuria.
Like this.
Me anak ibugin ka nga kapaninan roy rongo na.
is that what that is?
Anak karawat ya shampoo na ko yay.
Sure.
Kanga nakarawat ya kami depay na ko yay.
Yeah, that's fine. Jar 5. Yeah, it'll be interesting to see what we can get on that. Maybe if it's mucousy, we might be able to get some DNA from it.
Okay, this is the
Brown slime.
is yarn. So there's a, like, there are some rings that they can tell, like they can determine the conditions of the rock. Yeah.
There's
Is
what?
that some rings on the
There it is.
rock that can determine the conditions of the coral?
On the rock?
Yeah. Like by looking at the rocks, you can also identify like by if there's some rings on it.
If there had been a coral there before.
Yeah.
So like yeah, I mean if you see if you see like a coral face, but there's no coral, then we knew a coral used to be there. Is that the
And
on
those
these
rock
rock
can be
conditions,
determined as
environmental
well.
conditions. Oh, okay. So like sometimes yeah.
Yes.
There's a question about with the rocks, can we determine or see environmental conditions like we can with corals? So there are certain corals that if you run analysis on them, you can determine how old the coral is and what seawater was like when that coral was growing. And kind of you can do that with rocks. You can age date the rocks based on the amount of crust that there is. So how much crust has formed over the rock. What else can you do with rocks, Steve?
Come up, just try it.
You can. What have you said so far? Age the basalt. Look at the chemical composition of the crust.
Can you determine anything about the chemistry of the water?
That's a good question.
I know that other piece is pretty solid. It's neat gravel.
I don't know. I mean, you could look at how water interacts with the minerals in the basalt, how they alter over time. That might be interesting. They actually create new minerals over time if they're exposed to seawater. Yeah, I don't know. I don't know if crust or the rock actually captures
Any
any like signal of.
environmental change?
Yeah.
Yeah, I
I don't think we need any more, guys.
Yeah, might need another piece, unfortunately.
I think there was enough in there. We don't even know what it is. I mean, I don't want to.
Sure you don't want it?
We've come this far, Steve.
All right. You guys are just making
I was
more
gonna
work in the
ask.
lab for us.
No.
I want to ask you.
You want to sit at the library a little bit longer, don't you?
Do you want to stay here and I'll log because you know what you've collected and what you haven't?
Yeah.
Does that sound good?
I mean, so we've actually just transitioned. We've lost almost everything we've collected, and it's
Okay.
now totally new stuff.
Okay.
But I'll stick around for a little bit. Yeah.
Okay. I mean, that way, then we
Yeah.
can let Brian have some more time off, and I think it's good for you to kind of be in the seat for this. It sounds like, too, we're probably going to come up. I mean, if we're sticking to schedule, it's going to be off bottom around 7 anyways.
Okay, thank you guys.
That's
You
all
guys
I'm
like
saying.
answering your questions.
You can do it if you want. I just want Steve
She
to
wrote
say
it in
I
there.
like having the YouTube question.
Okay, or we can switch out, yeah. Okay.
Yeah, I mean, I could sit in back or I could sit up front, whatever you want.
Katidik, if you have more questions, you feel free to
Huh?
I don't want to deprive them of the Brian and Alexis show.
They
write
might-
them down
they're
again.
already- They're over it. Let me get my water and stuff.
They said you have a good question.
Yeah, maybe that would be a little bit less cohesive. I'm stumped, honestly. I think it might be a type of sponge.
There it goes.
All right. Thank you. Yeah, I wouldn't even have thought to have poked that, but I'm not so sure. Glad we did now. Manage this
What do you say?
unknown skin.
Science
Rock
without
skin.
understanding the unknown.
Yes, and you can get after the RV Falkor.
Yeah. Get in line. Yeah, this could.
Steve-O's
I was gonna go with sponge, but you know,
It's hard.
who knows? Yeah, I mean, it
It's
looked
not
leathery to me.
It's not gold.
It's not what?
It's not gold.
Not gold.
We're gonna go with gold.
It's not like raw, you know, gold foil.
I think it was that gold foil stuff.
Yeah.
Almost certain that was gold. Another stop. Another one of those bamboos that we see up there.
Can I tilt up and a little bit? Oh, there's your Narella. Narella something. Maybe we zoomed on that already. Yeah, so now we've kind of like, whoa. Everyone's gone. Okay. Nothing. I thought I had the B team behind me here. The next team.
That's not nice to call Tony and Jim.
Don't get me wrong, I feel good about it. Matter of fact,
I don't know what y'all talking about.
some fans will learn them, right?
It stands for admin.
It's fans.
Oh my gosh, I'm famous. I have fans.
Maybe that from
Yeah, this is I mean, we're not seeing like crazy big coral colonies, but this is just enough flow, and the substrate is really cohesive and cemented enough to be of pretty good stability. Very little sediment.
Okay, it's time. We have
What?
6 more minutes.
Yeah, almost there.
For those who are from Kiribati,
Ah,
there might be a new ship coming in, so if you have any other questions, just write it in English. Thank you. Sa Benini po.
you ever seen one of those?
Stop crying?
No.
A rose?
A plastic bag?
No. Is that a tunicate?
Yes, it is.
Is that?
Not tunicate?
It's called I think it's called a horsehead tunicate.
Wow, I've never seen that.
That thing, that was awesome.
It's so long.
I saw one for the first time last year, and I was like, this is trash. This is
It looks like a
this is
windsock.
garbage. Yeah.
Subsea windsock. You know, where the current's going.
Yep. Yeah. They're there for us,
It's
obviously.
It's like one of those weather rocks.
Massive tunicate. It's the Goodyear blimp.
Yeah.
Do you need a laptop? No, I have mine. I just put it somewhere. Thanks.
Have a good share.
Don't get
Thanks,
sucked in there.
Annamarie.
And it won't come out.
What's in there?
Another dimension.
I feel like I can see the RV like shrinking and going woo.
Magic school bus ROV.
Yeah. Oh my gosh, can we make that series? Magic ROV?
Kind of looks like
Wow.
one of those parade blimps. Like a
Are you logging that? Because I'm not.
I am. Yeah. Very nice tunicate, stock tunicate.
If we grab onto it, we'll know what the current is. Before we go.
Just hold it up.
No problem. crinoids and barnacles. No, yeah, this is. I don't think these are predatory though. I think they're
Just passive.
planktivorous.
Yeah.
Probably pretty old.
Do we have tunicates on non-associated? Yeah, we do. What'd you say it was called?
Tunicate. I think it's called a horsehead tunicate.
Horsehead.
Or something like that. Might be closely related. This one. This is maybe it's not a horsehead. It's something similar related. This is what we saw last year.
Where'd you see it?
I think it was off of the west coast of California.
Okay.
But much bigger.
Yeah,
But
that
I
one
think
was-
it was different than the horsehead that we saw.
That one's like on the sediment.
It's on the bottom, yeah, but it's really long.
Oh, okay.
Yeah.
Oh, yeah, there you go.
Biodiversity. That's right about right.
Yeah, I think that was the first one of those we've seen on this expedition. So we've done a half science watch change. I'm Alexis Weinig. I'm with the U.S. Geological Survey, and I've joined Steve as he's going to stay on as watch lead for a little while longer. This is cool looking.
Yeah, I feel like you pick any one of those rocks and break them off.
Do we need more rocks?
No.
Okay.
No, just commenting on the texture. We got some rocks. We're kind of doing stratified sampling, 3,300, 3,000, and then maybe one more before we come up. Nothing too crazy.
Little arrow worm.
Acorn.
Acorn worm, sorry. Well, aren't they also called arrow worms? Colloquially?
Those are the Kittignaths.
Oh, okay.
I don't think I've ever seen them on a rock like that. Kind of weird. I don't see them on straight,
Yeah, yeah.
flat surfaces.
Yeah, you don't see them like climbing.
Exactly. Yeah, it's kind of weird. Oh, I'll see something amazing, Steve.
It takes a lot to, if you haven't noticed, it takes a lot to amaze Steve.
I know.
Literally everything we saw
Like, you gotta
at the
think about what
beginning of
these
the
are.
dive.
Wow.
Yeah, like I couldn't put a pteroral into a family earlier.
Did your brain
couldn't.
explode?
No, I just picked it up.
Must know answer.
Must identify.
I didn't expect to do this much sampling. I didn't think we would see very much, but it's actually
Yeah, we're doing
pretty good, yeah. And
awesome.
I'm wondering if it's because we do see a lot more biomass, generally in the deep ocean, shallower along the equator, where we are right now. We're at 1 south, and we suggested in 2017 when we found higher counts of corals at northern sites within the Phoenix Islands protected area that it could be due to higher productivity in this area. And I mean, that productivity is going to make its way down this far, so maybe that's what's fueling this deeper, higher diversity and abundance of coral sponges.
These are very angular rocks.
All reminds me of just like collapsed layers.
This makes me think of like an amphitheater, like Red Rock Amphitheater. You know, like you could just set up a stage. Everybody can, all the little deep sea animals can come and watch a underwater concert.
Like shrimps in the band playing.
Shrimps in the band. Trying to think. I think this is the deepest I've ever been with an ROV. I don't know. What was the deepest we did in Pippa? Like 20 something?
26?
Six?
25? Just somewhere in there.
It's the deepest Sebastian's been in a while.
I know Sebastian's rated to 4,500, but do you actually ever go to 4,500?
Yeah. Yeah. Whenever we do sea trials,
Oh, okay.
we'll drop it all the way down to 4,500. But it's up there for a bit. But Mariana's Trench is definitely 4,500. Above.
Yeah.
Do you carry more cable than that though, right? Yeah, way more. That doesn't even look like you put a dent in that winch.
No.
Plenty there. Is it starting to look familiar, like your the start of your H1783 dive?
Does it? Good. Yep,
I
it
don't know.
does.
I haven't looked at the video yet.
I
but-
have to say, I would have to look back to remember that dive. I don't know how you
biggest
do
regret
it.
of the trip so far is not bringing those hard
Yeah,
drives.
actually, I agree. We normally bring everything. I don't. You have? Do you have all of those yourself?
Yeah.
Okay.
I knew I'd be accessing them, so I just brought them.
Yeah.
I can send you a copy
Or dirt.
if you want.
No, it's just dirt.
I think there is some of it that I would need for. We should talk about the scleractinian paper before we get off.
I got you a cup coral.
You did.
There were 2 cup corals.
Are they different
Not
or
me,
the
but,
same?
like these guys over here.
You.
They're predecessors, but they can take the credit.
Steve, yeah.
Steve, you're now driving the RV and using the arm and doing the science commentary.
He's pretty much amazing.
It's pretty much.
Pretty much amazing.
He's a triple threat, if you will.
No, because I knew you'd want the tissue. So, and I want the skeleton.
I know we
and
can't
you
share.
Can't have everything.
Here we got a chimera. Oh no, it's not. That's like a. Looks like a large Nazumia.
Oh, throwing out fish genera.
That's like the only one that I know from
I don't even know.
working with Andrea, and I'm probably wrong. What's next to? Oh, a sea cucumber.
Yeah, I've been having to look up the fish genera, and then actually the YouTube chat has been on their game today with the fishes, so don't steer me wrong.
Oh, I
Where's
gotta
the
hit the fish button.
Fish button. All right. Apparently the what we were calling the Basozetus with the these tendrils is actually Dicrolini.
I don't know if I've
Maybe.
seen that before.
We saw this earlier today. It
Okay.
has these like tendrils. It was probing the sea floor.
Oh,
Although we saw a little bit deeper depth than I would expect to see. So maybe it's different species or different genus, but very similar looking. Pretty steep here.
Where are we on the track?
So waypoint
Oh, yeah.
3 there, I think, is 2888. And then this is kind of the steepest portion, then we'll kind of level off again.
Okay.
I'm going to leave, probably find some mud, and I'm going to come back on the rock.
Excuse me?
No, kidding. The mud has been the best place for associates
Really?
of corals, yeah.
What have you collected today?
So we don't have many associates, but the one associated organism we found with a coral was a sea pen with a stereoschema, and
Okay.
almost every single sea pen had that stereoschema,
Huh.
and they only occurred in sediment. But we have a pair of cup corals, some sponge, pyrostylid, a couple rocks, a couple of
There's a
carcigorgids,
little
a couple of
sponge.
primnoids, and one black coral, and a bamboo,
Okay, yeah.
and 2 water.
You've got quite a little assortment there.
Yeah. But it's not, yeah, it's not many associates.
Yeah, yeah.
So
You
it's
haven't seen any crabs?
one crab, free living, not associated
Okay.
with anything.
Lots of those tunicates. Is that a sponge?
The purple one.
Yeah.
Sea cucumber. Tilt up a little bit. Yeah, I think there's a pretty clear transition here at this site between 3,000 and 3,100. Almost everything turned over. The one exception was maybe Pleurogorgia. We saw a
Mhm.
couple of individuals across that boundary. They may be the same species. Maybe not. We'll see. I think we collected one from deeper that looked more brambly than these nice arcs
Yeah.
that branches create.
Fish.
Yeah.
You're doing just fine coming in, face it.
I think it would almost be as informative to have
This little
a guide to the
jellyfish.
fishes based on just the shot from their behind as it would from the front, because most of the time all you're seeing is the
Yeah,
behind.
it's hard to get to the front. Oh, that's cute.
Second one of these we saw.
Yeah.
The one a bit deeper was a bit more vibrant red. This one's pale.
It's like almost yellow. There's a question about who is on watch for ROV. We have Cody and Jared on watch right now.
Hey, Rob. That's a very dark.
So dark, yeah.
Wow, cute. collegiate ones? Or do you like it? Sounds like all good.
I realize we don't have a jellyfish button for non-associated inverts.
Oof.
Good thing I can edit that right now.
Something over there. See it? There might be another one of those tunicates actually.
It is.
Yeah.
Waving around.
Oh,
There's a
yeah.
big guy. Is that a sponge? No, it's
Tunicate.
Do you remember how deep you were when you saw it on Nautilus?
Oh, it was shallow.
Oh,
Yeah, not shallow. Probably less than 1,000.
mm.
Different species for sure.
Like how in our non-associated inverts we have random crustacean instead of just crustacean. Like, what would that mean? A lobster?
Just
Like an actual lobster?
there are many crustaceans that pick one randomly.
Fish?
Halosaur out there somewhere.
Where? That? Oh, it's, yeah, it is. I think you're right.
Fish bun.
I did, I got it.
Oh, okay. You want to help me shop for cardboard boxes?
Sure.
I've had these tabs open for weeks. Alright, so almost at waypoint 3. It's pretty much right on.
Are we on
right on
schedule?
depth. Schedule? I don't know. Let me do some math here.
Maths. Math it up. An anemone. A new paper just came out about anemone distribution. Did you see that on Twitter?
No, it's not.
I'll tag you. I'll send you another Twitter present.
I like presents.
So this is our deepest dive so far on this expedition, and in this region also. We haven't been below 3,000 meters on any of the previous ROV dives in this Hallen and Baker area or in the Phoenix Islands protected area just south of us. So we're getting to take a look at a portion of this habitat that we didn't have any information before. And Steve's been on watch for most of this dive and collected a number of species or a number of corals and sponges that we hadn't seen before or would possibly be new range extensions. So interesting contribution to the overall story of what we're finding out here in the Phoenix Archipelago.
So the math is not exactly precise, but somewhere around 11:30 to 12 o'clock is off bottom.
That's not what
That's
we're
not very-
shooting for.
Has the dive plan changed since being
That's what
issued?
the
Maybe we can have that into the
yeah.
calculation form. I can work on that on my off watch.
Crinoid.
Dark crinoid. Definitely very different substrate here.
This has
Small.
changed a lot.
Yeah.
I've been on the ship for 4 hours.
Vertical wall, sediment.
Yeah. This
Got
pebbly.
everything.
So weird.
This place has got it all.
Hmm.
James, is that bathymetry here? Is that our mapping?
Yeah, this is like
Okay.
you see this section. Discovered?
I see. I see.
That's what we got last night on the way to the site.
Yeah.
You'll fit it. Put a camera something
Okay.
that looks on there. Yeah, it might be something. Right there. That's should have had. Oh, those good. Flash.
Fish. I feel like we have actually seen a surprising number of fish.
Yeah, there's quite a bit. Way more fish than actually in the mid
Right.
depth range. Yeah, it's one of those spinning. Yep.
Oh,
there it
yeah.
goes.
Do, 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 do.
He is on the move. Yeah, you know.
Burning some calories right
That is. That's one way to move. Yeah.
It is. They have such beautiful shells. You never really see them on hermit crabs. Like, you don't see a hermit crab with that iridescent shell. So they must. I don't know. He's done.
He's done.
He's tired.
Just like that. That's cool. That was the first one I saw, right?
Second.
On this dive?
On this dive.
Okay.
But we've seen other
We've seen
ones
them
also.
before, yeah. They never seem to be associates of anything. They just are on. Have you seen them on a coral or sponge? No.
It's a big. That might be a big spot lobster up there.
Ooh, yeah.
Big white spot lobster.
That's a percentage down there, right?
Mm-hmm. Yeah, we'll image this, and then there's something off to the left I want to get a quick zoom on. There's another snail. They're
Oh,
all
yeah,
over-
yeah.
all over the place.
Is it a squat lobster? Or one of those? Wow.
You like balance?
Yeah, sure, why not? We haven't done one in a while.
Is Tim around? Did you see this before?
I don't. We haven't seen this before. This is a new Munida maybe, but
I've never seen this crab before.
it's big. It's a big crab.
There he is.
There he is. There he is. Yeah.
Look at there. He
Look
is
at
right there.
that.
Look at that thing.
You got manual.
What do you think of that?
No, what do you think of this?
Have you ever seen this?
Chunky squat lobster.
No.
See the little parasite on him?
Where?
Between his leg right there?
Oh yeah, maybe. Yeah, right.
Or it's like very red
They do
color.
that.
Yeah, I don't know. He's a big boy.
It's got very short arms.
Yeah.
Super short.
They gotta get hauled in there.
I would love a suction if that's possible.
It's really big though.
Is it really big?
Is it really big?
so
We'll put it in a
there's,
bio box.
You got lasers?
Need lasers?
They're just coming on.
We could get the snail
Just
too
need-
while we're at it. Oh, there's two snails.
It's like
Did you image that snail before it started rolling?
no, but we can get it here.
Curious, yeah.
Wow.
So it's
It's a monster.
it's like
I mean,
14 centimeters.
That's this big.
Tip
It's
to
not
tip.
monster.
Tip to tip.
Oh, that's not that bad.
Will make a great photo.
But it's
for
not
my
a suction sample though. Then.
Well,
We could
do
Suction and then suck and blow.
We could put him in there and just shake the bio box.
Okay.
All right,
Let's
it would
go.
be great for the profession.
There, good job.
You want to do some
Can we
images
image?
first?
Let's image the snail too before we do that.
Look at them. They're all over the place.
You wanna keep the lasers on or off?
One,
Only see
two.
one. Okay, never mind.
I see two.
By all over the place, I mean there's two.
I
but
was
you
like,
know,
where's
It's
the-
a
yeah, I was
real
like, where are they?
abundance.
it is an abundance.
Steve, that's very good eyes,
Those
but I
are
was like, I
really
can't
good
see
too.
them.
Oh, I need my glasses on. I just realized that I don't
They
have.
have
them.
eye spots. The genus name for these snails is Gaza, Gaza. Like they're
They do have pretty shows.
yeah.
Really nice.
That's full zoom. Yeah, I'm sure there are videos of these on YouTube somewhere.
Yeah.
If you want
Must be.
to see more.
It's nice.
Was there one other thing?
Before we do that,
I think it's a
no,
crinoid.
I'll come back to that later.
So if that crab moves, it may move away from it.
Yeah, you wanna
It's gonna
come-
go. It's gonna go backward.
Do you want to do in situ photos here?
Yeah.
I don't know. Tim's got
There we go.
special
Are you
sample
all
in
the way in?
situ.
No, I'm max zoom right there.
All right. Beauty.
Frame grab. Frame
Click.
grab.
Beautiful.
Beauty shot.
It's a solid
I'm very
monster.
interested in his little claws compared to.
Boxing gloves.
Short arm, right?
Yeah.
And it hit you with the rostro if you get that close.
Do you have a jar big enough for this?
Yes, of course.
Okay. Oh. Just checking. All right.
Let's see what
I think
happens.
we're ready to go. Here, why don't you sit down for the sampling? You want to do
Yeah,
that?
you want to get out?
Uh,
Doesn't matter to me.
I feel like this could take a while.
I don't think you will.
I'll stay here.
Look at all those labels I did.
Look at you.
All right.
I'm excited for this one.
Well, that's important.
It is.
It is important. No, this is so. This will be really good for our comparisons of
Yeah, it's a
certain
very
comparisons we're trying
different.
to make to what we other crabs we see at shallower depths.
We don't see very many that are these that live among the rocks versus on the coral. So it's probably a different type of crab than we've been able to collect so far.
Got it. It's crazy. Just one of these just sitting out like that,
I
right?
know.
You know, but it has been that kind of a dive right down here. You get not quite singletons, but few. You know.
Yeah. Pretty sparse.
Yeah.
I don't know, but this is like Snail City, though. So,
What? With those two, you mean?
yeah.
Okay.
Well, three. I mean, if you count.
We saw one already.
I wondered. I thought that was the one that rolled down.
No.
Oh, that's a different one.
Different one.
It is Snail
Practically
City.
gathered.
They're everywhere. Did you see that thing up there?
Hmm?
Did you already see that thing up there?
That is a xenophyophore.
That white thing?
Yeah.
Oh. He snugly pulled the gun out.
Do you think it's a type of immunida?
I think that's a Galathean crab.
Oh,
That's
okay.
A Galathean. It is bigger than the suction hose, isn't it?
That's a good shot.
This could either work
That's a good
splendidly.
image on
This
there.
could go really
Yeah,
wrong.
it could be splendid.
This could go
or-
wrong and viral.
So you're zoomed in right there.
Are you making labels as you are collecting now?
No, we just
Well.
pre-labeled the
That's
FK21.
That's a good
What
idea.
if?
All right, so
You're getting efficient.
I don't know how they go.
Oh, I don't know if he's gonna fit in that.
Don't go into it.
Wow, that's amazing.
That's pro.
That's pro. It doesn't happen every
It's
day,
not in-
folks.
It's not in the bucket yet.
Nope, nope.
Nope, not in yet.
Where are we going? So it's in the bow box, so it could.
Yeah, whatever you
These
want.
open.
Then we
Yeah.
have 2 boxes open.
Right. Okay.
Now, if we open up B, that means you cannot open up the spot box again.
That's right. It's a one-time thing.
A one-time use.
What we got down here?
Y'all are okay with that?
Like you can't open the other
Can't open
chamber
up any-
either. That's what he's saying.
We're losing a buy box spot.
We're losing about 2 of them.
Unless you want to put it with the Chrysler Gorge, there's no
Put it with
associates.
Chrysler
Can we,
Gorge,
we put it in there?
Of course.
Yeah, because then we'll have two bins then.
Plus this one goes
All right,
fast.
what am I doing?
We're gonna gotta be ready to.
You have the next number ready? It's
Oh.
maybe you have to go back on the last page. I think it's 450. 450 is the next number. I'll double check.
Yeah, there we go.
Well, it's not in there yet, so that's just.
Section's off.
Section back on for a second, and get in a little bit further. Okay.
Bow box open up a little bit.
No, that's not right. Here.
41.
There. 450 is the next one.
450.
Okay, close.
Put the bow box.
Trying to get this angle a little bit there. Okay, section off.
Section off.
And
tail is up in there.
There it comes. What you got there? One of your R-letters popped over in this direction.
That's okay.
Okay. Try to whip it. Little hard.
This might be a push cord. Just put a push cord cap at the end.
Here we go. Here we go.
Oh, there we go.
Close it.
Amazing.
Oh, now he's like,
Don't eat my crazy board yet.
Oh, he's trying to swim backwards. That's all she
Pop
knows.
up just a little bit. I got my hand on the trigger.
There we go. Little shake. Oh, come on. Thinks it's in a cave now.
Created the perfect squat lobster habitat.
That's it. Nope, see, every time it comes out. Good job. All right.
yes!
Wow.
That's what
Okay.
I'm talking about, bro.
There you go. Way to go, Jared. That was awesome.
FK 21 450.
It's 450, yeah?
I'm gonna lock that bio box.
Good idea. That's Bowbox C.
Buy box C and sample 450. Monster. Already on the coral.
Oh, I can't wait. I can't wait
All
to
right.
see it.
That was a thrill. Thank you, Steve. Next amazing thing.
Yeah, sure.
Do we want to suction in one of those snails or? Okay.
Yeah. What are you gonna
Yeah, so they opened
do? What?
Quiver 10 to, or like way earlier in the dive, and like shiny stuff, like shimmering water came out at 10. Was there any already?
No.
It might be
It's over
just
here.
temperature. Was it?
Could be. I don't know. Maybe it's fresh water.
Yeah, it's also yeah.
You guys fill with fresh water?
No. Not out
I
that I
think
know
that
of.
number 19 up or that one right above 19
Maybe they did. I mean, they fill them, so.
See, do you want a snail or
That's
no?
okay.
No,
No, it's not.
thanks.
So what?
So
I saw some math over here.
we're making very good time. I think.
What is that local?
Head off here in the bed. I'm gonna try to disable this thumbnail.
Mhm.
That was exciting.
Okay, cool. I was disabling it so nobody opened it. Make sure this one's still working. That's working. Yep. Okay, cool. Alright. There we go.
Ready.
49.
No, it's okay. I gotta go view it. I don't need to look at it anymore.
You going?
Keep going.
Hannah. Okay. Here we go.
Kind of interesting that it's on this kind of featureless area. I thought it would, even crabs would appreciate more rugose terrain.
Yeah, or places that they can hide.
I suppose, for a crab, if you were a crab's height, this would be like
It's pretty rugose.
pretty rugose.
Mm. Sponge.
Do you have Slack up?
Yeah.
Just in case this was. Actually, I see something here. Lower right corner is translucent tentacles thing.
Oh,
It's a very, very fine
crinoid.
crinoid.
It's clear.
He is.
No pigment. So it's
Or like white, sand colored.
Yeah. Okay. Onward.
Carry on. There's a little stalk up there.
When I was looking at the video from 2017 and from 2019, I could hear the sound of the joystick here.
And now you
I was
know.
wondering, what is that
Yes.
noise?
If you control the thing with more gentle hands, then it doesn't click though.
So
Yeah,
that's
I-
what
I know.
You can really
I know. I
control
was
the thing
commenting
and-
on others
Oh,
Yeah.
because when we listen to the video later, you can hear the clicking.
yeah.
The issue too is I try to go really slowly and then it wants to jump.
Yeah.
You ever do that?
No, that doesn't happen to me.
It doesn't have. It doesn't have. You'll notice that.
I always notice the chairs, like you can hear the chairs creaking,
Yeah.
like you know when the ship is moving because the chairs.
That's true too.
Okay.
I don't know.
I mean these ones maybe, but other ships you can really hear it.
Right.
You guys have well-oiled chairs.
Federal.
It's on the checklist, the pre-dive checklist. Oh, there you go.
Is there a pencil here too? I think I have the label pencil. Not a
Did you lose your pen?
Oh, no.
Rocks here.
This is the second one I've seen.
I got just my pen here.
What happened? We had like a nice maroon
Scab, too.
pen.
Oh, I don't know.
No, like over here.
There's they're all in the basket. And
Critter
around.
camp.
We don't have percentage on here either.
Yeah, asteroid.
Oh, okay. Well,
Alright. put your name on it. And,
Awesome.
What is it?
you can install Critter Cam.
Critter King.
So our
Can I have
goal.
a piece of gum?
our
Oh no,
goal
I can't. I can't. That's gonna be annoying.
Our goal by
In
7 o'clock say
2.5 hours.
is to do
Oh wait.
get up to 2,500 to 2,600.
Where about is it? What point?
That is. I don't have that plan in front of me. Do you have that
Is it
plan?
here?
No, it's not been printed.
What? Okay.
I've been looking at it on my computer.
Well, I can find it.
It should be waypoint. It's pretty close to the next waypoint because the last waypoint is 22.
2,500.
So now it's pretty much the next waypoint. All right.
So what we could do is like we can jump the ship a little faster. Move them a little faster, and I can put a little more steam instead of just going this slow.
Yeah. I mean, I think it's doable. There's not much to zoom in here, but
And is it a huge deal for 30 minutes on the other side? I mean, let's say it took us till 8, is that a huge deal?
let's aim for 7.
7?
Times 7 to 7:30. Yeah.
Okay.
7:30, right? Or 7.
Yeah, 7. But I mean, if it's a
Let's
little.
aim for 7.
7.
To get to that depth range, or whatever else we get is bonus. That's the cup coral we got.
Oh, is it?
We got 2 of them. I don't know. I haven't seen this one before. It could be. So there's one.
It's a very flat. Is it very
It's
flat?
very flat. Yeah. One of them. One of the individuals. One wasn't even attached. It was just kind of sitting.
Oh, interesting.
And the other one was like a pancake, just barely attached.
Okay.
Like a pedestal thing like this.
Yeah, that's like that little one we collected shallower. It was like that too.
Yeah. Yeah, I mean, half the things, like the shallow stuff, I just have no clue where it could be assigned to. Cucumber? Yeah.
Looks like a cube. Cube.
Yeah. Cucumber.
Ooh.
So it looks like after this we're headed to the Winslow Reef area, which is a shallow water reef in the Kiribati waters, but we're going to be staying on the U.S. side of the Kiribati-U.S. boundary. There are some fairly shallow banks in that vicinity that were more than likely, in almost certainty, above water at one point, possibly forming an island or a bank.
What is that?
Or
Is
reef.
it just something in the water?
Anemone.
This.
Oh. Goo? Gelatinous. Oh, maybe it's a Pelagothuria carcass.
Is it? Oh, it does look like goo, right? Mucus.
Deep sea mucus.
Deep sea
Somebody's food for a whole
mucus.
year or more.
No, that's good.
So we'll be transiting down that way and maybe doing some dives along this shallower feature. We're going to map it first. We have some partial mapping which we'll use to plan our first couple dives, but we're going to try and map the other side of the shallow bank. The summit is at about a little bit shallower than 300 meters, so quite shallow, and that will be some of our shallowest dives after Howland and Baker. So going from the Abyssal to the possibly mesophotic between today and tomorrow. We should have, next bet should be, will there be photosynthetic algae?
At what?
At the summit.
I
At
don't
the
know.
summit.
It was like 260 before.
That's pretty close. We'll see.
Little hermit crab on the run.
I'm always amazed by how slow everything moves, but hermit crabs always move the same speed, whether they're
Yeah.
in the abyss or in your rocky shoreline. Very energetic.
Hermit crabs are one of those groups that really go all the way across all the depths.
Did you put lasers back on? Yeah, it's interesting. I almost feel like while the life down below was pretty sparse, there was actually more of it than in this depth range right here. Maybe it's current related, but it seems like the current is fairly similar. What's really striking is that we had a lot of corals and a lot of coral diversity when we first set down at 3,300. The current was stagnant, and here at least there's at least a little bit of current, but maybe it just doesn't support the larger corals that we've been seeing shallower. One of our deepest dives until this started somewhere in the 2,800 meter range, and we were definitely seeing much more stuff. Not a crazy amount of stuff here though.
Tried to cover it.
I'll skip the cup coral. Go for the fan.
Got a lot of.
Not gonna go full in and out.
Yeah, that's good.
Oh, wait a second.
What?
Probably saw something, but
That little tiny sea star?
I thought it was on a stalk or something.
It is.
Is it on a stalk?
Yeah, it's on right.
Focus.
Thanks.
Is it a sea star?
I think it is. It's very
Can we go
little.
look at that?
You want to bring down the
Oh, it looks like it's eating
It's on
in.
the carnivorous sponge.
Bring down the ISO. He
Oh,
said.
wow.
Yeah, let's go over there.
It might be eating a arm again, like we saw that little one eating the bracingid arm.
Yeah, I'm not sure what it's eating. To me, my money is on sea star eating carnivorous sponge. But it's in a crevice, so I have to go take a look.
It
But
is
this
in a
might be our
crevice. It's very small.
deepest known ivory of some type.
Can we get a little bit better look at that?
We are.
Okay. Because I think that's a carnivorous sponge being.
That's what Steve just
Okay,
said.
sorry.
No, you're okay.
Transit from one room to the other.
Yeah, so we saw one of these carnivorous sponges deeper, but this one is very tiny, and it might be some of the deepest evidence of ivory. Once we know what it is. And furthermore, it looks like a goniasterid star. It might actually be one of the deepest goniasterid observations in this area, since we don't often see them below 2,000 meters or so, or they're very poorly known. Looks like what it is. That's what I would say.
Yep, I agree.
Want to suction sample it?
Suction sample both.
Yeah.
Let's do a photo first.
Okay.
Mm-hmm. And get lasers on it. We'll come out and get lasers, yeah.
I did a frame grab.
I'm gonna pull out and put lasers on it.
Okay.
Alright, hit it. Do we have lasers on or am I come out more?
Oh, lasers on.
Yep.
Just turn them on.
Size of the laser.
Yeah, so you're looking at like something with 4 centimeters in tip to tip. Alright. We can go for collection into a suction jar 6 or whatever's queued up next.
Okay. I had it in on 8, so.
Oh wow, we have a lot of suction
8
samples.
has some stuff in it, but that's
Yeah,
just from
flush.
that's a flush one.
Yeah.
I wanted to get as much suction for that option as possible.
So 5 has the skin.
Okay. 5 has the what?
He called
You
it the
didn't see that?
skin.
That's in it.
The rock skin?
It's not skin. It's just.
It's maybe a sponge.
God,
Might be a sponge,
that
okay.
was terrifying.
You scared me just a little bit.
It has the what?
Go to first section. Oh yeah. I forgot that's what I was gonna do. Up and down.
See all the things you miss? You need a microscope to go through like some of this terrain because it's like there's
Yeah,
stuff on
honestly,
all sides.
if we hadn't stopped for that cup coral,
no, we would have never have seen it. So this is 5451.
Okay.
Okay. Put down home. Ready for a second? Go ahead. Hurry. Zoom out.
There you go.
Got them both. One. I don't know if you'll be able to see the sponge with so fine, but I'm guessing it's in there.
Q what? I mean, sorry,
6.
jar.
Jar 6. 451.
Good old fashioned look around.
It's a goniasterid.
That's just getting
It's a goniasterid.
Yeah.
So cute.
You gotta feel a little bigger.
Yeah, it could have broken apart too. They're pretty sticky, but
Might be.
yeah, I'm pretty sure it's in there.
Down to number 7.
Okay. Can we zoom on the coral over here? We don't need to go over there. Do they need fresh corals for the experiment?
Let me see.
And this might be a
Yeah.
Norella worth sampling since it's one of the bigger ones we've seen. It's got an oath on it too. The first ophiuroid that's not been associated with a sea pen as a dive, and one of the first ophiacanthids we've seen.
Mm.
Shrunk looking too.
There's a little snail down or something. See the shell?
Yep.
Solitary hydroid.
It's coming into focus. Yeah, a lot of this white zoanthid
Mm.
as well. It's the only one we've seen really. Alright. Give it another minute.
Okay.
60 seconds. 30 seconds.
Like 60 seconds is a long time. There's another big
I think we'll be seeing
acorn
more.
worm.
Yeah. Oh yeah.
So do you want to sample some of these or no?
Maybe. We'll see some more.
Onwards.
We had hydroids on that. Zoanthids.
We had a lot of primnodes yesterday.
Yeah.
A whole lot of them.
A whole lot.
I know y'all's that's end of the dive was like
From notes.
pew 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 pew.
That was some of the highest coral densities. But that, I mean, that's exactly like what it was on Titov.
Right, on the other
Nautilus,
side of
yeah.
Titov, yeah.
Which is why it was so impressive. And then it's really remarkable how sparse it actually was for 90% of that dive, and then just the last 10% was Coral City.
Yeah. Yeah, it was not
Geodes.
Coral City at 4 a.m., I can tell you that much. What do you want to say about geodes?
No, guyots. Maybe they have a. I mean, that's going to put guyots as an outlier for coral density. Whoever looks at the coral counts at that site.
You mean when you look at coral counts at that site?
I'm not making any assumptions.
I want to go back and look at the dives that we did see in rubble, but not large live colonies like that one, and see the depths that we saw that.
Yeah. Yeah, I mean, I feel like, so I have some interesting notes on that from Jarvis that you might find.
Oh, okay.
Oh, another one.
Oh, look. It's his friend. That's good, though, to see another one. Lets us know that they're probably all around.
So helps us sleep better at night.
No.
Still not doing anything, just kind of sitting there. I thought they would be feeding on something. Sometimes you see them feeding.
Or maybe
But
it's taking a break.
What's interesting is their carapaces look really fresh. So like,
Yeah.
maybe like we saw a ton of barnacles down here.
Oh, goodbye.
Yeah. Yeah, he learned from the last one.
He saw his friend in the box down there.
Hey, Jim, why don't you keep on moving, buddy? Get out of here as fast as you can. He's cruising.
There's a question, Steve. Have we observed sea stars eating carnivorous sponges before?
That might be new for me. I haven't seen that. Definitely not out here.
Do you have any theories about why crinoids become detached from their stalks? Do you think something tries to eat them?
Yeah, I wonder if it's predatory. I don't know. I mean, they're supposed to be like ancient, like
Yeah.
extremely old. So I don't know. It's the same as the last one. Keep going. Umbrella.
Oh, I did not log that last immune crab. Oops.
Yeah, stocked crinoids are actually some of the most well-preserved invertebrates in the fossil record because of these ossicles that make up the stalk and then parts of their body.
Yeah, they're pretty hard-bodied for being invertebrates.
Well, we've had some dives that have many of them, but not at the densities that I'm familiar with. Like, we've seen them crazy dense at certain other sites.
What is that? Oh, that's a rock. I think it looks like a dead sponge stalk, yeah.
That's big. Huge. That's probably where it came from right there.
Big anemone.
No.
Seen probably thousands of them on back so far.
Thousands of what?
Cenophyophores.
Oh, yeah.
They were really abundant, and probably one,
That's
two,
a dead sponge.
maybe 3 or 4 from that I could identify different morphologies, but I'm not an expert on those.
Found something. There's no expert on.
It's every rock.
No, this is. I feel like this is my backyard. I mean,
Yeah.
I spent 3 years of my life practically just searching nothing about the deep sea in this part of the world.
That's really awesome.
I still have no clue what's going on. No, it's not true. I'm learning every day.
Yeah, lots of xeno, but therefore.
We don't have to stay on the line. We can follow the rocky ridge. Yeah. We lost all the sea pens. around 3,100 meters, 3,050 meters, something like that. Yeah, CTD temps only come up about 0.1 degrees C, but I don't know. We should be moving into the no. We're still a little deep. We're still probably in the deep water. This does put into perspective, though, when we drop in at 2,500 meters, we drop in at 2,800 meters. You know, we think it's barren down here, and everything below that is kind of the same, and that's not true. There's much more zonation on the seamount if you do more exploration, if you do more, spend more time there. So this will be very valuable. I'm very interested to see what kind of specimens we're bringing up. Particularly those pernoids.
Yeah. Little tripod fish. We're really cruising.
See if there's any rocks or air formation. Go.
Let the vessel move
Yeah.
out a little bit.
Go explore. There's some sand and some caves.
Every time I was about to start into my little spiel about how diverse deep sea sediments are, we find rocks. So let me know when you want me to start that.
Go, go, go. I want the spiel.
I'll tell you as soon as you see rocks on the sand.
Flag right there. Good rock.
Is that the equivalent of a handbrake turn?
That was.
An ROV?
100% a handbrake.
That was pretty impressive. I've never seen that done before. Oh, these might be whale scours again.
Really?
Did you see those?
I didn't, no.
Whale scours are there? They were very clear. These are less so.
They were
Yeah,
very
these-
clear.
Huh?
They were very clear.
Very clear. I mean,
Bless
not,
you.
not like recent fresh, but you know they were filled with some debris and stuff. But usually, if they're parallel to the slope and not perpendicular,
Mhm.
I mean, if it's perpendicular, it could just be slumping of
Right.
the sediment. But yeah. They were a bit deeper than this too. These are pretty shallow. I don't know. The whale shark was pretty cool, but for me, if we saw a beaked whale diving down a
Oh my
depth,
god. Yeah.
that would be literally groundbreaking.
Would that make you like scream in excitement?
Would it? I don't know.
Jared over here knows how to speak deep whale. So.
Oh yeah. Would you mind reaching out to your friends for us?
Just turn on the speakers and the ROV.
Yeah.
They're on right now, but
Y'all have to understand that Steve and I have been working together for the last 6 years, so he's very used to me poking at him about his behaviors.
We'll try and put up the bathymetry again in a few minutes when
James gets back.
James is still on watch.
Yeah.
Yeah, I'm just worried that like we might be headed for more sediment between here and here, and then it'll pick up again. So, oh, what is that? Oh, crinoid. Crinoid or umbellula?
Oh, I didn't see. They do want more.
Trying to find
More
frogs.
Yeah.
Narilla?
Pushed away a little bit.
I'll grab the next one.
Right, those rocks kind of really disappeared on us.
Very complex xenos. So is it that different species? Is it species of xeno that create different structures?
I think so. Yeah, I don't know, but never really dug into the xeno
Yeah, me neither.
biology.
Anyway, I created a waypoint by mistake. Every time you guys move my waypoint, it comes back.
Arrow or a acorn worm.
That's fine, Tony. We're having the same issue down here. Every time we move it, one of the other screens goes black. We have a little glitch going on. But you can see your waypoint still?
Yeah, I've got my waypoint up here. It's just,
Yeah, no problem, brother.
Cheers, mate.
Check that thing out real quick. See if that's rocks.
Just checking it. How's it all going?
I'm just trying to move past this little bit here.
Uh-huh.
But
You're doing really well on our track.
we are doing very well, yeah. We're gonna be on track to. I think we should be passing over some of Nautilus's start waypoints.
Can you click for a second?
Just that black
Cody,
little
to
mushroom.
the left, that coral over there.
All right. Well, great. Thank you, guys.
Yep.
Actually, that looks like
I'm
a
gonna tilt-
live
I'm gonna
version
tilt
of
up
one
a
of
little
those
bit, yeah.
sponges.
Spoke too soon.
Oh, yeah.
Ficus, yeah. All right, you want to go for this one?
Yeah.
Okay. I'm gonna sample this one.
I mean, it can go in a quiver too. It doesn't have to go, in a dye
quiver,
box.
yeah.
We have a large sponge back there, which we'll take a look at after we take a sample of this coral.
I think this is gonna be one of the large polyp marilla. There's a couple of species that have just like disproportionately huge polyps.
You collected one primnoid super deep that had huge polyps, right?
Yeah, I wasn't sure. So the one primnoid we collected, this one, had very, very small polyps, but they
Oh.
were pairs, and they were very weird morphology. This one, where is it? This one, unknown octocoral. I couldn't tell if it was a primnoid or chrysogorgia. It has characteristics of both, and it was really just,
Is that the one with really big polyps?
yeah.
Okay.
Yeah, like really, really big polyps. Alright, let me go take a look first and get some imagery, and then we'll do basically 4 of these branches, snip off the top.
And
Yeah.
a lot of barnacles on this rock too.
Alright, do you want to do in situ?
Yeah, give me 1
out
second.
Here?
Your manual?
Hold on, let me.
One,
Yep.
going in. Alright, snip, or oh, a lot of focus there. Alright, you got it?
Yeah.
Okay. I think we're good to go. Can we get lasers one more? Just
Lasers on?
Yeah, lasers on the colony just to get an idea. So it's like 25 centimeters or so tall, something like that. Yeah, so basically 3 or 4 of these branches, 10
Yeah.
centimeters each branch, something like that.
I'd go for those 3 that are kind of clumped together. Yep.
All right, so this is going to be 452.
Okay.
About 10 centimeters, you said?
Yeah, I think so. That's fine. Marilla species. Think we need more?
No, no, I've
Oh.
never seen them open a quiver with the sample in hand. Those are big polyps, wow. Four, 5, 2.
I hope those polyps aren't too big to get into the jar. I haven't seen.
enough?
I think that's
Yeah,
enough,
that's good.
I haven't really looked at the proportion of those respiration jars versus
I mean, maybe they can, probably fit a whirl of those.
one whirl. Yeah,
But there's like, what, four polyps in a whirl?
Yeah, probably 3 or 4.
Quiver 15?
Quiver 15. Yeah.
Jared, that was great how you opened the quiver with the sample in your hand. I've never seen that before.
Birching. Slow down the fortune. Fortune. Alright. Continue?
You.
Oh, there's that big sponge
Oh, yeah. Want me to look at the sponge?
that we'll look at. Yeah.
Thing over there.
It's associated. Is that crap?
Let's see. Let's see.
We did a little pirouette around this thing. It's pretty big.
Bring the ISO down some.
Which one is
That was a
down?
shrimp. This. Oh, bring your.
Left is down.
Right, I think. But I don't think I'm controlling it.
Wow.
Wow
That
is
one of the
correct.
This thing is
biggest.
huge. Look,
one of-
look at it in front of my porch.
Look at the. Oh, yeah. Yeah. And this 10 centimeters is like.
So one of the biggest sponges. I think it's Caulophacus, but I have to check my ID guide again.
Looks like an elephant here.
Can we take the lasers off? Thanks. Lots of little. Either little snails or little hermit crabs on there, too.
It almost looks like a work of like abstract art.
It does.
So that's what these always have reminded me of.
Look at it. It's unreal.
Yeah.
Big white blob.
Well, and we've been seeing those big, tall stalks, and it might be that the dead stalks, which might be what these are, what those stalks were before. For some reason, they died or detached.
Yeah. Something probably in the, I don't know, on the way out, can we turn lasers on one more time? On, yeah, just to get it on this side of it.
It might be nice to do a come out of it with the black background, with the lasers off, to do a bit of a beauty shot too, because this is probably the biggest, bestest sponge we've seen.
Charismatic megafauna. Yeah, it's probably 60 centimeters across at least. Probably more though.
Lasers off or on?
Let's go off now.
Yeah.
And then Cody, if you want to just come out some when you are leaving, so we can get that more big picture.
Zoom out just a little bit.
You.
Oh, I didn't know I was in. That's full wide.
Full wide.
Yeah. It's just that close.
Okay.
Yeah.
This?
What was that you just collected?
Oh, the coral? Narella.
It's for
It's
Randy.
For experiment.
It's one of the large polyp Narellas, yeah, for experiment. I don't, it's probably one of two species. do we want to do? Dinner rotation.
Are you hungry? You want to go eat?
Yeah, I kind of go
Yeah, go.
eat something. I'll come back up and hop in after that.
Okay.
You do a mosaic. A single thing, right? But, I don't know. What do you think?
Do you want to sit?
Yeah. Sit if you want.
We don't have the base of it. Back off.
We're talking about mosaicing this.
I don't know.
Okay. Shrimp.
Yeah, there were 2 shrimp on the back side of it.
I think you have to look at the back side like this.
Yeah, see them? There's 2 of them.
I just see 2 shrimp there. Could be more.
Wow. It does look otherworldly.
Did you put lasers on it?
We did.
Massive.
Yeah, it's like 60 inches across.
Wow, that's cool.
It gave me like an idea for a new medical product. Lung on a stick.
Yeah, looks like a
Just
lung.
like
Lung
walk
on a
around-
stick.
Walk around with a lung on a stick. Is it, so that's a sponge?
Mm-hmm.
Mm.
That is really, really. Look at the structure.
Yep.
Mm-hmm.
Gosh. Have you seen that before?
Not here.
Like that.
Ever?
But we've been seeing a lot of the. We have seen.
I've seen other lungs on a stick, but not like that.
I haven't seen that sponge before.
We haven't seen them that big
Say.
before, probably.
Yeah, you shrink that down to something.
Yeah.
Maybe it's okay.
But we've been seeing a lot of stalks that have fallen and dead, so I would hypothesize that it might be from those types of sponges, and for some reason the big sponge gets blown off.
Yeah. Okay. What are we hunting for now?
We're just going.
I saw that Narella, I thought it was a little, and a bunch of different branching. I mean, the angles of the branches look weird to me. Maybe I was far away from the monitor, but anyway. Oh, switching palettes right now. Okay, I'll be right back up. Sorry, no, me. That's all me. Alright. Let's roll.
Let's roll.
Somebody's sunglasses.
Steve, he always has them in here. He's too cool for school.
Too cool.
Too cool for
For glasses.
ROV.
Right.
No, not true. Nobody's too cool to work with an ROV.
Alright. 28, 23 meters. Should be coming up. So we
Got
wandered,
about
we
an
wandered
hour
off
and a
the line
half.
a little bit.
Yeah, because it was all sediment on the
Yeah.
line, so
Yeah. That's cool.
we got a fish.
Great. Another stalk sponge. Perhaps a small cousin.
Smell, yeah.
What depth did we start at today? I'll make a look at that.
30 something.
I was sitting right here when it happened and I can't remember. Here.
Yeah, when did this pen run away? You ran away with it?
No. I don't know, or it disappeared between on the way down. Is this not it?
No, I liked this one.
Yeah, I did too. It won't be over there. So. We started on bottom at 33.57 meters. We've come up 500 meters. It's pretty good.
You're good. I don't think we see it down here.
See,
we're
this
just
one
checking
of
out
those
this ridge
stalks.
here. Thanks, Tony.
Yeah. Patchy, huh?
Mm-hmm.
It's like a low relief little sort of a mound here with
Do you want me to go over the hill or
rocks on
stay on
top.
the ridge?
We'll stay on the ridge. It's parallel to our line, right? Our track,
It is.
more or less.
Oh, there's a big.
You always
Whip?
stay on the ridge. It's a massive whip. Does it have polyps? What are you gonna say? It's bamboo whip?
That's what I'm gonna say, but we have to look at
Let's
it
have
first.
Have a little. Always like to guess.
It is. You can see the nodes now.
Right? Do you see a crabs on there? Because if we see crabs, we got work to do. I'm gonna let Adam, you gonna fly this?
Yeah, you want me to start bottom, top bottom?
We're at the top now. Do that. Be great. Just a pass here.
Looking about, it's definitely over 3 meters.
Wow.
Yeah, about 10 feet. We're gonna fly it down though?
Yeah.
Okay, I'm just gonna go in a little bit. Oh, sorry, you can hear the noise. I wasn't supposed to do that. Okay. Is that in focus?
Yeah, that's pretty in focus. Now it's not.
Coming in.
See him closing up?
Because of the pressure wave?
Yeah.
Look at this. Not the pinnules, but look how
Spicules.
Thank you.
Yeah.
How strong they are.
Or
Polyps coming
sclerites,
off all
sorry, they're
sides.
not
Sclerites come off all sides. You know, polyps coming off all sides. Oh my God. Look at that though. They look like they would hurt if you grabbed it. Polyps, were they all in when we came up?
Not all
So-
in, but they definitely, there were some
They
that
feel-
were out.
They feel we are here.
Yeah.
That's cool.
Oh, there's some that are still.
Nobody else living here though.
Nope.
Nope.
Can you get one close up of the polyps before we
can.
leave?
Alright. I'm gonna zoom in at him if that's okay. Just when we get to the bottom here. Well done.
Yeah, look at all the, and the ones that are closed, all the sclerites
Yeah.
along each tentacle.
Yeah. Look at
That's
here.
pretty unusual.
This is? This
Yeah.
is it?
Like just to see them all, like they're, it's not the
Right.
real spiny ones, because sometimes they have those. Ooh, that was cool.
Whoa, that was cool.
Sometimes they
That's how
have
they do
big,
it.
sometimes they have big sclerites that like poke out of the polyp even when it's closed. And these, there's such small sclerites that they all just close with they're around.
Yeah. And they're all pointing down.
Yep.
So that makes them
What?
downward facing bamboo.
Sure.
Anything on that? Nope. Okay, great. Thank you, Adam. That was nice.
We collected one bamboo today so far, I
You
think.
did?
I think so, yeah. Branched bamboo.
I missed the spongivory.
Oh yeah, it was like this tiny, it's a tiny little sea star on a tiny
Did you
little.
snip it and get it?
We slurped it.
Yeah. Okay.
It was on a tiny little carnivorous sponge.
We're supposed to be coming up a ridge, you know, at the top of a ridge, but you know. See, it sort of dips down and goes back up again.
I'm gonna shoot for more northern heading and try to
Yeah.
get back up
So you
that
come
way.
back on more to the line. Yeah. We're 28, 22. I think our deepest dive was 24,
26, I mean.
24, like 60 back in 2017.
okay.
But you're right, on this cruise it went deeper. Closer to 26, I think. on
Yeah.
this cruise. Yeah.
But this is by far the deepest dive out of any of the cruises.
Mm-hmm. So what's your impression of the dive that you've seen so far?
I can't wait to get that crab up to the surface. That's all I'm thinking
Did
about.
you see a second one?
Yeah,
That very large Galatheid crab,
yeah.
white.
You saw it swim off. You saw it swim
did you
off?
see- Yeah, I didn't see it swim off.
Yeah, when we approached that one, it swam. Good.
So we're probably gonna head into a little depression here before we go up slope a little more. There's no depths here. Let's count. Okay. All right.
Just cruising along until we get to the next.
These little sea cucumbers on the floor.
Yeah. Well,
Yeah.
that's right.
Roger that.
No, yeah.
Will do.
Oh,
No,
there was a couple sea stars. If we see a sea star, we'll take a look. But not for the sea cucumbers.
Yeah, looking on that.
Oh yeah, look.
map there.
So like right there was one. Just
Saw
keep
one?
an eye. Yeah.
It's just below us.
If you look down the camera, you should be able to.
Yeah.
There it is.
You look down, Tim.
All right.
Yeah, we'll take a look. I just saw like 3 or 4 of them.
You think it's on something?
No, I
This
just
is what
think
we
it's
saw
good
this-
to take a look at
Snaps
them. Yeah.
We saw this one earlier today.
Oh, we did?
Yeah. It's a goniasterid. If I could focus on it. Come on. Come on. The world's waiting. There we go. What a beautiful focus that is. Yeah, we saw this one earlier. I thought it was feeding on something. There's something small there. Do you see that little purple thing?
Yeah. A little worm or
I don't know what it is.
copepod or something.
Tim, I'm just gonna just slide
Go-
a
go.
little bit.
Yeah, I'm gonna back out a little bit.
See if you can get it over top.
Okay.
Look straight down on it and wrap it over there. I know.
All right.
Good picture.
Okay, here we go. Nice. There I go.
You can do it.
Thanks for
The encouragement.
the encouragement. So I thought I saw this thing on the move a little bit, but now, yeah. Thought it was moving to the right a little bit. But we did see this earlier. I don't think
Okay.
we didn't
No,
collect
no.
it, right? We got a good ID. It was so up off the seafloor, I thought it was feeding on
Oh.
something, right? Thank you, Adam. Line back up here. Good. There we go. I think we saw that quite early, like it's 3,200, something like that.
Oh wow. Was it on sediment or rocks?
Sediment. It was just on the seafloor. It was rock, I think. Sea pin. I guess we've seen those today.
What's that out there?
What?
It's a fish.
The white thing?
The fish.
This thing here?
Yeah.
It's a
Fish that's sitting on the.
Sitting on the bottom. Ooh, maybe it's a bathysaurus. I love those that sit on the bottom. The
You
triangle
can just
head.
take a zoom on it without.
Roger that. Gonna go in for a little zoom?
Yeah, we can just slide down here next to the boat.
It's a bathysaurus. He shoots. He's cool. That's fabulous. These guys have like a golden eye and really sharp teeth. If you go around the other side, you'll see the really sharp teeth. They're sit and wait predators.
Are these the lizardfish?
They are called a lizardfish. Yeah. See that you can see the teeth now. If I can get ISO focus in. Come on. You drive it, Adam.
I'm fine, you got it. It's all you.
Yeah. I think I might come out just a little bit.
Yeah, come out a little bit.
Trying to sneak it out. I think I see a little copepod parasite there. Something near the fin. Shifting position a little bit toward us. Go to the right, Adam. Won't see the. Nice. What's that?
I think
Okay.
Brian
Yep.
and Steve are gonna come back up when they're done. So yeah, thank you though.
Can they come down a little bit? Okay. Right.
There you go.
Just hold it for a minute.
Not there yet. No, but I think Brian and do you want to?
Okay.
Brian and Steve were gonna come back after that. Okay.
Do you wanna go eat?
Yeah.
Hold that for a minute.
Okay.
Just gonna grab my phone.
Oh, sorry.
You can log on there though. That's fine. Hey, friend.
Hey. How are you?
Good. What is this fish?
I'm zooming on a Bathosaurus.
It's very cool.
Very cool. These are.
Back. You wanna go eat? Or you can stay there. You go.
You wanna go? Go ahead.
No, I'm good. Right? They'll just sit there and sit there and sit there
It's very calm.
until something comes by.
Can't believe he didn't take off when he landed.
Didn't do anything, right? Okay. Great. Thank you. We saw a cloud of smoke earlier, but in one of these things, when they take off, they can really brush the sediments. Okay. I guess I should write some stuff down here.
Just
Here, do you want to? I would say 7.
Okay.
Frankly, I mean, I think.
yeah.
Oh, I'm not getting better focus here. There we go.
All right.
9:12.
Thank you.
Yeah.
Earlier Steve was saying that some of the big marks are saying
So we're seeing whole thurians and sea stars out here. Not much in the way of ripples or anything fast-moving currents.
Nope.
So you feel for more fish, but some fish love the sandy type bottoms.
Absolutely.
Do you wanna?
Now I'm gonna
Do you wanna
click for
draft?
1 second.
at 7 hours, it's looking more like even at the farthest point, it's gonna be 6.5.
Perfect.
So if you're going closer, it's gonna be just shy of 6.
Brian, would you mind taking over locking here?
Yes, we had 7 feet.
Yep. Okay, so that was the farthest point. Okay. Yeah, so that's, let's see, is it, it's like 65 miles or what?
65, yeah.
So,
So then what we're kind of have to do is not, now we're going to but it will be more time, then we don't need to have this one. We need to have as much of this as we can.
yep.
Okay, we're at a depth of 2818. Cruising along to our next waypoint, back on the waypoint 4.
Chop down the angle.
So we thought there'd be a sandy region of this dive like it is right now, but more of a depression, even low, and there's an acorn worm on the bottom there on the left, I think. And another sea cucumber, a holothurian coming up in the middle of your screen, but we should be in this for the next, looks like, I would say probably 300 meters with this.
That's
So
fine.
we'll see.
We can do whatever it takes, an hour or two, and then we.
And so I'll dive plan, so it takes us 6 hours to get there. We leave
Yeah,
here
that's 65, 8.5 hours. So,
4:30, get on the beaches, then should be ready to drive by 6:30.
So today, so far, we've made 20 collections of things that we think may be novel or new range expansions, deeper. This is a deeper dive than we normally do. We're currently at 2,814 meters, but we started out much deeper at 3,200 meters. What?
Hey, can I just fill you in? You weren't on.
Okay. I'm just talking to everybody.
yourself.
I am. That's what old men do. They
Eat dinner.
That's all right. Well, they're coming back, I think, right?
I think they are. As soon as there's one more person back, you and I can go to dinner.
All right. Oh, you should have it.
No. I'll wait for you.
No, no,
I'd
okay.
love to eat with you. I'd love to dine
Sea
with you, my
cucumber.
friend.
Dine. Tonight
Here it
we
is,
dine.
dining.
Yeah. Cue the whale shark. Just gonna keep it up a little bit. See these little depressions, these linear depressions like that?
Yeah.
I've seen these in the Gulf of Mexico and the North Atlantic, and some people suggest that things like this, maybe not these, but things like them are actually sperm
Beaked
whales
whale
that come
scars?
down.
Yeah.
They come down and take a chunk out of the seafloor. feet on feet. This depth is not outrageous, actually. Can you believe that?
How long does it take them to get down? That's one thing I was trying to think about before. Brian and I were chatting about it.
I don't know. I think there's some estimates on how long it takes, but there are certainly in the North Atlantic, in the canyons, they're diving this deep.
I was still trying to figure out how much time they'd have on the bottom. That's why I'm asking how long it takes them to go back down or not.
I don't know what their
know
descent
either.
rate is, but
Look at all this delicious food.
But they
Why
know
would-
they have tagged them, so they must
Yeah.
know what it is.
Well, that's a good idea. Look at that. You preemptively answered the question.
What?
So good.
What do you say?
Somebody asked, what are the ditches in the ground? And you just answered it.
Oh yeah.
So
Well, I
perfect.
don't know if that's what they are, but they're awfully conspicuous and don't see who's making those. But usually they're a little bit bigger than one like you see right here. There are certainly gouges in the sediment of the seafloor. This could be a lot of things here, but the ones I've seen are much bigger.
You just don't see any creatures here that would have made this, right? This is the thing. It's like, and certainly nobody caught red-handed.
You know, in general, I don't see a lot of bioturbation. There's a sea cucumbers here that are turning up, taking in sediment, squeezing out the organics, and digesting it, and then they just release them back. So I'm not even seeing any trails of anything. Yeah. In the Gulf of Mexico, we saw, less shallower than this, we saw circles of holes as the fish through. These perfectly symmetrical holes that go around in a circle. Had no idea what was making those, couldn't figure it out. We would see crabs and shrimp sitting on them and whatnot, but it was really, I think in the end it was a fish that was doing it.
A fish?
Fish was going up, making holes, and moving on, making another hole.
Huh.
Ah,
In a perfectly symmetric pattern.
crazy yeah, I mean, it was a perfect circle.
Wow.
Amazing.
That is amazing. You know, I think behavior is really hard to observe down here, right? So you just see all these like signs, and you have to play the
Yeah,
game
that's
of
right.
the guessing game of how did it get there and who did
That's
it
right.
and why did they do it?
That
and
infer the process.
How long has
just-
it been there?
Just from a
and-
flyby, you know?
Yeah. You're right. So, Rowan, Brian, and I were chatting about the putative beaked whale ditches. You know, he brought up the point that some of these could be really old and they just don't get sedimented over that quickly.
Yeah. So we're on top of that ridge. It's become quite sedimented, hasn't it?
Maybe. We don't
Yeah.
know what's over the edge.
No, we don't.
Never do.
Just beyond the lights.
Hiking up a tiny trail.
Here we go. With a match.
Oh, even better.
Yeah, right. It's not quite that bad, I guess. Another sea pen.
Oh, here, I'll write that down. How do you read my mind like
It's
that?
worth working out.
It is.
Is that a polycule? No. All right. So we're generally headed in a northeast direction. Top of this ridge at 2793. We just broke 3,000 meters. Coming up. I mean 28.
Is that waypoint purple now?
Hey, Jay. Doing well.
Think? That's what I'm gonna think. Not purple.
Shrimp
Okay,
and
what
oil
is it?
thurians
Click
and
it.
sediment.
Did it go black? Yep.
I like the magenta up there though.
Beautiful. Sounds good, brother.
Can't miss it.
She's
no matter what you do, it kind of puts us in this neighborhood.
By the way, I think our Ascent's going to be not 2.5 hours.
Because it's either we get in here, which is still, I think it's okay. So it's or
See, I think this is too much.
2.5 hours to get on deck.
Well, we're at 27 now.
Oh, okay.
Oh, I see what you're saying. You're saying an hour, 1,000 meters. It's 28 divided by 60.
You think I can do that in my
Come on
head?
now. I don't have my iPhone with me.
Here, I got it. What do you want?
2,800. Well, no, do 2,700.
All right.
Divided by
45.
No, that's not right.
The captain just did it. He thought it was 2.5 hours. Even if it's
Really?
2 hours. So let's say it's this.
Yeah.
Then it's 3. Then it's 5. Then it's 6. Then it's 6:30. And yeah, I mean, that gets us there.
Yeah.
I mean, that's the whole
Anyway,
thing.
I mean,
Yeah, let's go.
you can do this, or you can.
No.
I'm all.
Which is fine.
I got it.
I would actually be very happy
Okay.
with where we are.
I got it.
Okay, so if you are listening to us and we keep turning off the mic, it's because we are planning. We're planning. We're plotting and planning. Is that what
Yeah.
we're doing? We are scheming even.
Oh,
man, there we go. Wow. Right then and there. I told you we never know it's right over the ridge.
No.
We're trying to think about, so we are on day, I don't know how many days we're at with it. This is dive 14 of the expedition.
Yeah.
And we only have so much time. Time is a zero-sum game when you're out here, and we try to make the most of it. And so we're really enjoying this deep dive. But we know what we have coming up, which is also going to be pretty exciting, which is to go to a seamount that tops out at about 300 meters, where we're going to be spending a couple of dives in a row. And so after we leave here, we will be headed over there. And so we're trying to figure out how much time to allocate to each dive. So anyway, you guys are along for the ride, which we really appreciate, and we will be back in the water. We're not done with this dive yet, but we are not going to stay in the water for too much longer. But we will be back in the water roughly 11 hours from whenever we leave the bottom.
Sounds good. Every time I go up, I lose the white balance.
That's over.
Yeah, there's a big sea cucumber.
27.75.
And a stalked crinoid.
Yeah. We're not seeing much else. Have a look, see?
Very
Like that
cute.
with a sail on the back. You can see the intestines there. That cylindrical tan you see is actually the digestive system. That's literally seafloor sand going through the animal. It goes in one end and comes out the other.
Just waiting for another organism to eat it.
Oh, yeah.
Sorry. It was right there. There's a question about how long it takes to surface from this depth. It'll take about 2 hours. I love those yellow stalked crinoids. They're beautiful.
They're great.
When was the last time we picked up a rock? That's where we're at it.
I think we have a few today.
We have 2.
Maybe not recently. Is that all? No.
We have 2. Should we? Would you like another rock?
Yeah, before we.
Yeah, just we have a perfect rock field right now.
Yeah, anywhere.
All right, J-Rod.
15
centimeters, nothing crazy.
What'd you say?
10-15 centimeters, nothing crazy.
Okay.
So you say smallish? Is that what you said?
No, 10-15 centimeters, yeah, nothing crazy.
Give it a little poke, Steven, and see if
Yeah, let's go rock poking.
Is that like cow tipping?
Let's see it, sure. Looks good to me.
take it in situ shot.
Yep. Yeah, let's go in. Nice lumpy bumpy.
Bumpy.
Alright. In situ 1. Do 1 with lasers. Sorry.
Yeah, no, you're fine.
Lasers, yep. Alright, lasers in situ. Or, I mean. Alright, let's go. Says 453. Botryoidal rock. Can you, half a botryoidal rock?
Similar pieces now.
Okay.
Broken half. Right?
Maybe that, right, yeah, right there.
What about this one?
Yeah. I mean, the other one's fine.
The other one just broken half. It's right. The one that we still want.
I get the feeling that this is all kind of just crust. I mean, yeah, does that one move? Right behind the jaw? Nope. Yeah, that could work.
Pulling up the big rock.
Pick it up and then lift off. Yeah. That'll be perfect, yeah. I guess I should hit one in situ one more time after comment out the other ones. Round with
Alright, so that's the half from the first one that broke. Yeah.
yeah, I think that's okay. Yeah, let's go for that. Just how sure are you?
I saw
Pretty
it.
sure.
I saw it.
Okay.
I saw it.
That looks fine. Then.
So that's the half from earlier?
Yeah, half
Okay, so cancel.
half cracked.
Good with that piece?
Yeah, that's fine. Looks good. Should be easy to identify in the basket. Great. Good sample and.
You gonna check it for another piece or?
No, that's all right. If I see a must-have rock, maybe.
453.
1453. Yeah.
1453?
Oh,
453.
That's what I wrote. 4.
Okay.
5 3. Yeah. It looks like all of this just is like sheets of crust, so I'm kind of worried about there not really being a lot of rock in here. Trying to think about how to package these couple of these most fragile corals before the next shipment comes up. I don't know. We have bubble wrap, but like these would be even crushed by bubble wrap. Some of the precious corals.
Yeah.
I don't know. Other than putting them in like a Tupperware.
I mean,
Permanent
that's
storage
the way to do it.
with bubble wrap. Yeah.
Just put it in a hard container, but then they're going to even flop around within the Tupperware potentially.
Just like put some bubble wrap in there,
Yeah.
maybe, or packing pillow. Any no predation there? No. Basket now. Oh, what is that?
Sponge, I believe.
Sponge. Furriates we were seeing. I think it actually might be stuck on and just kind of died there. We never actually got any bits of those Furria. I don't know if that was of interest to you. It was kind
The
of
one you were breaking apart?
no. That was something else.
That one's dead, so I don't
Yeah.
think that's worth grabbing.
No, the other sites, we saw a bunch of this Furria,
Yeah.
and it was one of the more dominant sponges.
Keep going?
Yeah,
Yes.
yeah. We're just discussing. The problem with Furria is it's hard to collect whole, 'cause even when you dry it or preserve it, it just shatters into a billion pieces. But a genus level ID, I guess, is better than nothing. Furria is very something that might be in the EX collections, I think.
How's it going?
Just picked up a rock and we're trying to make way. We're entering some of the steeps here.
Trying to make it the way we were, right?
Yeah, thereabouts. I mean, that's about the depth range that overlaps the previous dive at this feature.
I wonder what causes these things to die.
Sponges?
Yeah. What is the mortality event that triggers death? Is it predation? Is it age? Is it change of food supply?
Yeah. Yeah, I mean, I'm not anticipating taking it in more collections, unless you see it as something phenomenal.
Okay. Like that little bitty sponge.
Yeah. I mean,
I'm joking.
we got some sponge, but I mean, maybe.
What
I don't know,
equates phenomenal to you?
I don't know.
Some little caves.
Yeah, I think. I mean, I pretty much checked all the boxes. The only other thing that I think would be useful would be, you know, we're light on sponge collections generally. So if we saw Faria, I would probably get some Faria, the frilly
Yeah,
looking glass
okay.
sponge.
It's not what you just said. It's really hard to preserve and it turns into dust.
It does, but
Okay.
what are you gonna do? It's a sponge. Yeah. No, I mean it's possible. It just it doesn't do well when it's dried. You have to
Yeah.
put it in a hard box. Yeah, but I mean, there's plenty here. This won't take as long as we think because there are hardly any associates on this stuff. Yeah.
We have not seen very many jumbo octopuses on this cruise.
That is very surprising. Yeah, Helen Baker was in Jumbo City in 2017.
And we saw a lot of them at Pippa.
They all hatched and flew away.
Do you have any desire for more Niskins?
No,
Okay.
unless you happen upon a coral garden.
Those are pretty.
Did we get enough water for Anna to do some microbial stuff?
In fact, they're both for her. I don't really.
They're both these.
They're both for microbes. Yeah, I don't really steps.
Oh, here's this coral coming up.
There you go.
Yes, sir. We'll take a look at this.
Probably some crabs in there.
You wanna have like a close look or just like
Probably a close look, because if there's a crab inside, we'll be more interested in it. It's very pink colored Christgorgia. There's definitely somebody
There's
home.
one right there.
2.
3 maybe? Or a shrimp in there too?
Hey, reserve me.
Thank you.
You wanna name it?
Yes, please. Oops, where am I? It's waving at us. It's very dense coral, so it's hard to see this squat lobster. This might be what Tim calls the red face Europtychus, which it's just oh, that's a benthictinophore.
That's my guess, yep.
Roger that.
Definitely.
Tim would like the crap.
Okay. Where did he just call you from?
Randy called me from
Oh, gotcha. Okay, so we're planning to collect this with the crab. Looks like there's only one crab. There's a ctenophore up top, and then something else, but I think it's like a shrimp in here. That's okay. I got it. Thanks. It's just me being slow. Or maybe it's just like an amphipod. There's a solitary hydroid on there too. Okay, J-Rod. I think there's one. So this is a ctenophore up top.
Yep.
You see another?
So look like there's one there. And what's that right there?
That's a crab down below.
Is that a crab?
That's an amphipod.
That little red.
Oh, okay.
Yeah,
thought
I
saw
know.
a pond.
I think it's. It's okay.
So
Just below the crab.
And putting it in a Biobot?
Yeah, I think so. So Biobox C is locked shut. So A or B is fine.
Okay.
Did you get pictures?
You got a trail.
Where's that crab down here? What's that down here?
Oh, that might be.
I think there's a second.
There's
crab
a
down
second
there.
crab down there, but there's one right here, Cody.
Okay, so we'll go somewhere here.
Yeah. .454.
Yep.
Great. Before we take off, I'll just take a look at the coral again to see if we can see that other crab that's tucked in there. Thanks. Yeah, thanks.
I think you both.
Did I break it?
Click it one more time.
I think you both switched.
Oh, okay. No.
Click it one more time.
Yeah, there we go. It's so hard to see them through. Yeah, I know. Get some coloration, but just can't get a good shot of the whole crab, but that's okay. We did what we could do. We can carry on. That was in Biobox B, yeah.
Yep.
I think that was the first big, or relatively bushy chrysogorgia we've seen today. Got a sponge we can take a look at over there.
You don't want to stop and look at it?
No, I think we'll just zoom. Or if you want to, that works too. It's up to you. How's that, Brian?
Good enough.
Okay. There we go. Yeah, we'll take a. We can see what's growing on it.
Sponge
Looks
stock
like-
with hybrids.
Yeah. So probably like that big sponge we saw earlier. We've seen a number of these stalks left behind, so something causes that sponge to die or break off, and then the stalks just are left behind. They become habitat for things like the hydroids, or sometimes we see crinoids living on them.
There's a live version of that.
Yeah, actually that I think we would like a collection of. Wow, it's so white.
That was the first thing we saw today was this tricky spot.
I know. I like this one. And I took my vlog with her there.
No.
Right. I'll plug in some.
What do you got for us about this sponge, Brian?
Not much.
Looks like we've got some shrimp living in here. We've seen this sponge a couple times, but it's been dead. So this is the first really live version of this that we've seen today. And there's sometimes questions about why we say a sponge is live or dead. And when they're alive, they have this really, if it's a glass sponge, they have this white, almost bluish color with the lights on them. And then when they look dead, they're more sediment colored, mostly because they're covered in sediment by that point.
The thing that always amazes me about this is how little tissue there is actually. When you look at these up close,
Yeah.
the tissue is extremely thin compared to the skeleton.
Tim, do you have is Tim here? No. You want to just try and take a clump of it?
Yep, I think so.
I think we're just going to try and take a pinch of it.
Take a little pinch of it.
Yeah. can go into a quiver.
Go into a quiver. Pull the pinch off the top.
Yeah, wherever's easiest for you.
Where US DAS is?
Yes.
I don't think it's working.
Is that enough or you want more?
You can get, if you want to put that in the quiver and then get that one little piece that's already hanging loose.
Okay.
Alright. You can put it back somewhere else.
Yeah.
Little mice and going for a ride.
I don't know if that's a mindset. Oh, almost. Maybe he'll just swim into the quiver.
Okay.
Alley-oop.
Oh my
I
god.
don't know how you do that.
What on earth?
That's other, and like that's like you got like a seventh sense of where the manipulator arm is.
I just cannot even understand.
That just blow your mind?
Yes.
Yeah, I did. That's two for this cruise where I've just been like, how in the world does he do that?
Well, the
It's
other one was
J-Rod.
J-Rod, yeah.
Slip that aplicot
Oh, you're right.
coral
Sorry.
like it was a wedding ring.
Wow, that was amazing. Jeez and Pete.
All right, this is
Welcome to
455,
Team
right?
Texas, buddy. Welcome to Team Texas.
Thank you, Cody.
This is 455, right?
455.
In 16?
Yes.
That's where I was born.
I'm gonna,
Alright, good to keep
I'm gonna
moving?
leave. Can we,
Yes, good
Do you want to
to
zoom in
know.
on that sponge?
Oh, sorry. We want to zoom in on the sponge.
What do you want to look at?
What do you want
Just
to?
see what it looks like when you open one up.
Oh,
and
the
what-
damage
There's
section.
things in there.
This one's kind of dying off, it looks like, in the root.
Looks like a sponge.
Yeah. Yeah, okay. Nice. Awesome.
Looks like a loofah.
Yep. That is a type of sponge.
A loofah is a sponge?
I mean, I would consider it a type of sponge.
Oh, well. I guess I'm thinking of like synthetic loofahs. Oh, sorry.
Work me here.
I did. Sorry, Brian. Amazing manipulator work by Cody. We need to have a slow-mo replay feature.
Yeah, we need the da-da-da.
Yeah.
Sports Center.
That'd be a great highlight video for SOI to produce, is a highlight of best ever collections.
However, I expect us deep sea dorks are the ones that might
Deep
really
sea dorks.
and other RV pilots might be the only ones that really appreciate how much skill it takes.
Elephant tusk.
There's a piece of lava, like, the lava toes.
Yeah.
The ones that are, like, it hadn't sheared off, and so it was just this tube hanging out, and it's huge. And we free flew and grabbed it.
He's like, oh, y'all can't grab that. You check her like, yeah, we'll go there.
Oh, it's another one of
Check
your
out
little,
food in, grabbed it.
it's another windsock.
Oh yeah, man, we've seen a lot of those today. That's our third one.
The windsock.
Yeah, it's like we think it's a type of tunicate, but they're much farther off
You
the.
know which way the current's going.
We do.
Guess I'm moving over more than the last one.
We should just tie a windsock to the ROV, then we'll always know which way the current's going.
Or just hold on to one of these.
Yeah, but I don't think it would do very well going up and down a couple times.
Put it back down again. Put a rock on it.
I'm not sure what the white parts inside are. Don't.
Go next.
They could be.
Up there on the jellyfish, right?
Oh,
Well, what?
what's that?
Is that another one of these things?
Is it? Oh yeah.
I just, my prop bullshit.
Oh yeah. So two. Earlier we saw two snails, and Steve said we're seeing so many snails, so we could say the same thing about these tunicates.
Did you get 3D model this time?
I was just making myself a note to try.
I'll try to make it around without hitting bottom. You want me to do any more on it, or?
Nope, I think we should move on.
Keep climbing up to, what are we trying to get to, waypoint 4? Lots of these stalks up here. More so than we were seeing down below. The broken off sponges. I think there's a little, I'll just do a quick zoom. Sorry, a snap zoom. Little squat lobster in there again. Shrimp at the base. Looks to be a similar morph to the one we saw that we collected. That's good. Thanks, J-Ron. Oh, yeah.
earlier that wasn't in the
Here's
survey.
a siphonophore.
It's a big one.
It's a cool little cutout. I think we have possible corallivory right here. Take a look at that sea star.
Bushy tail.
I think this is our first instance of corallivory today. We have,
Oh, yours, your name is
thank you.
Dr. Millard?
Yeah. It's really clinging on there. Looks like there's a branch.
Slack is what you did.
Slack's always working.
Do you wanna see the other side? Where
Um,
it's bowing down?
Yes, but let me just look at this branch first. Oh, this
That
little.
wimpy branch?
Yeah.
That little guy, didn't he have?
looks looks like it, yeah, had all of its polyps eaten off. I bet that if it tried, there's no way that little tiny branch could have supported that sea star. He's a chonker.
I just don't know how negatively buoyant they are, though.
That's true. You're right. Shouldn't say that. Oh yeah, there we go. Okay. Now that does look like there is some stomach extrusion going on.
Oh, yeah.
Oh, he's chowing down.
Yeah.
Nasty.
That's full zoom.
Did you wanna be closer?
I think this is okay. But we are gonna want to collect this. Just the branch with the sea star on it. And the sea star. I flipped that.
Think you should be able to reach it?
Yeah. You wanna go for a collection now? Have the pictures?
You good?
Yep.
We are good to go.
Come on in. Block time. Alright, see what you see. Here you go. Where's that? Stuck on the rock. Stuck on the rock.
That's spongivory. So that's the.
I think the break there where it climbed up, ate, and stopped eating, and then got on that branch, and was only eating like one side of it. Interesting.
Yeah. It is strange.
Okay. It's like I've got a little lollipop.
Yeah, it is. Bloop. Great job.
Alright, 4, 5, 6 in equilibrate.
Yes. Well done. Are those tops new? They look shiny. Did you give them a polish?
I was thinking the exact same thing though.
Were you?
Yeah, it really was.
See?
Did y'all clean it?
Yeah. Okay, we can carry on.
So what was that? Well, I'm kind of missing the giant squid and the Great Pacific Owl. I just collected coral. Snips. You should have looked at these. First, I do that.
There's a cup coral. I think we've collected a couple of those today.
Did you see those cup calls?
We don't need to look at them, no.
I think you got 2 of them
We
there.
collected two, yeah. Oh, there's another one of those sponges.
Is it half dead?
I don't know. Let's take a look real quick. Does not look
Yeah, I think it's dying.
as healthy. Yeah.
The other one was a little healthier, right?
Yeah.
Agreed.
Oh, well, now it's a lot more white that we're.
Looks like it's gone.
There's a little black coral down there, I think, too. The other one had these little shells on it also. Some type of little gastropod. Jeez. Let's look at the. Oops, sorry.
Jared, if you can go all the way around, that would be great.
the shrimp again.
Can we go back around the other way?
Yeah, yeah.
Then I have to get to the other side.
And actually, if you want to back off a touch and get a little more, make
You can get this side?
Yep.
Like that?
I wonder if
Yep,
that's why
perfect.
they end up breaking off the bases.
Dying back.
Yeah, so then that's probably more brittle.
Well, that's interesting. I mean, these things don't have like any kind of circulatory system, so I wonder
No.
if they. I mean, we know they can get to like 10 feet tall.
Yeah.
But still, I wonder how they move nutrients around when you.
Somebody calls an apple.
Oh, shrimp's coming back.
All right. I think that's great. Thanks. No idea.
With that little thing on the.
I think it's just blowing. Actually, I don't
Oh.
think it's alive.
This is pretty different terrain than we have had. Lots of sediment drape here.
Still a decent amount of exposed hard grounds, though.
Yep.
We got a couple of things over here on the right.
Got some sponge. Can we take a look at these sponges? This is mostly dead. Let's go up.
Spongivory too.
You just want the white ones?
Where's sponge? Oh, there's a
Back right.
sea star. Let's look at this one first. Two very different types of, not very different, but two different types of sponges. Hello, sponge. It feels very close. Thank you. Big anemone at the base there. And again, where we're kind of seeing the base start to degrade before the rest of the sponge. Don't see any shrimp living on the outside.
It's a little rinse spot.
Oh, a little mysid, you're right. Cody's got the eye.
Being all up in your face.
Yeah.
You want to get a little bit closer?
No, it's okay. He's very tiny.
I got you closer.
I know you did.
Super tiny.
Yeah.
Do you want to
Oh, he's like, goodbye. Farewell. We can take a look at that sponge over there. With the big sea star on it.
There might even be two sea stars on it.
Oh. That would be very interesting. shrimp probably. Oh yeah, I think you're right, on the other side.
You wanna go to the other side?
Yeah, could we come around and look at it from the other side?
That looks like a different star. Certainly, it's not a star I've seen on a sponge.
This one?
The big one,
Yeah,
yeah.
no, me neither.
This is likely going to be a collection as well. J-Rod.
Did you hear Brian? That we'll probably want to collect that.
The star?
Yeah, and the top of the sponge.
And the biobox?
C.
And C is full.
No, that's not right. Oh yeah, two different little sea stars on here. One big sea star, one little sea star.
The sponge does not look healthy. That is not a star.
No, I
recognize
do
at
not.
all.
Me neither. Oh, sorry, I moved.
Okay.
Okay.
Those,
Oh.
some manual photos.
Thank you. Wow, it's so cool looking sea star though. Looks like a giraffe.
Is that a joke, Steve, or is that actually what it's called?
It's a joke. We have two sea stars on the same sponge. And the sponge, you're right, does not look like it's doing too hot.
This is the rare zebra sea star. Oh, we found something that Steve's never seen before.
Let's take a look down the rest of this. It's always interesting to me when they get to the top of something. Like, why wouldn't you just eat the bottom?
Doesn't really have eyeballs.
I mean, I know it doesn't have eyeballs.
Doesn't know when it's missing or how
But wouldn't you if you came upon a food source?
they. When it comes back down, it has to eat on the way back down.
I guess so.
Saving some for later.
I guess. Alright, you ready, Brian?
Sure.
Is that a yes? Or
Yes.
is there okay. We are going to can we collect so that we have both the sea stars? And put it in the bio box?
You're missing the picture.
I see that. Where did our tooth go?
I don't know.
I feel like we're sampling a lot more than we're gonna have a pretty full basket actually.
We've got something, yeah. If you need to move a little lower on the base, that's fine too, to get a better grip on it. it was a shrimp.
Inside?
Yep.
Yeah. That is a big sea star. Nicely done.
457?
Yes.
This is one of those times you put up the bat star and call Chris Ma.
Met star.
Do you guys want to talk about barra trawler?
About what?
Barra trawler?
I don't know what that is.
Pressure change.
Oh. Go ahead, Brian. Go for it.
Sure. So there's a huge pressure differential between obviously where we are in the deep sea and the surface. Right now, the pressure gauge on the ROV is reading 3,949 psi, whereas we're at 14 psi up here on the surface. And so most of the invertebrates that we bring up handle the pressure pretty well. They seem to be more sensitive to temperature. If you get a fish or something like that with a swim bladder that has gas internal organs, they obviously need some kind of decompression. But the inverts, really, temperature seems to be more of a barrier than the pressure differential.
Got some sea cucumbers.
What's that?
Looks like a
Some kind
black
of
coral. Yeah. That's a good shot of it. You're good. You're good. Okay.
Still a lot of xenophyophores.
So many.
So many.
I've been surprised by the amount of fish on this dive too.
Yeah, there's been a fair number for this, a lot for this depth.
Yeah.
feel like the fish hang out more in the sediment. So we're out on these more sedimenty dives. I feel like we always see more fish than when we're in the rocks.
Got a percingid just sitting on the sediment, which is also kind of strange. Ooh, a little bit of pavement.
So with the RTCM corrections for the CNAV will be in the same or would extract over from.
Yeah, the squat lobster also falls in the same category of the inverts. Like it'll probably still be alive when we get it on the surface.
It's tough with we are really deep, so sometimes like things shallower than 1,000 are pretty much always going to be alive when we bring them up if they're inverts, but it does get a little more dicey as you get deeper, so it's hopefully going to still be alive, but there are some differences in depth that we've seen too, just for certain types of taxa. They don't get distorted or anything, it might possibly also just be a time constraint. So when we come up from this depth, it takes about 2.5 hours, so depending on how long it's already been collected for, it looks like a dead sponge, then if they've been in the basket for a long time, sometimes that just impacts their ability to make it to the surface in good condition. Wow, it's a very long. Looks like a coral.
With one little branch?
With one little branch. Yeah, it looks like a bamboo. We'll just do a snap zoom. Snap zoom, fly by. Yeah, that works. That's good, J-Rod. Whoa, that's right there. There's this one little branch.
Must be a new species then.
Don't say that. Lots of sea cucumbers.
Tell this area sometimes has pretty
Yeah.
good currents with those bed forms like that. Another one of these stalked tunicates.
Yeah, man, they're all over the place here. Also known as a windsock to J-Ron.
Deep sea windsock.
Or current sock? Why are we calling it a windsock?
Current sock. That's fair.
It's a current sock. We don't need to spend too long on it though. Goodbye. We are moving relatively quickly right now. We're getting towards the end of our dive time, so we'd like to cover as much of our track as possible. So we're kind of moving and zooming at the same time. There's another
current sock.
Current sock. Very botryoidal here. Lots of small bumps on all the rocks. It's a little glass sponge. No, it's okay.
Dead stalk.
looks like we have a note back from Chris Ma that I think the larger sea star is a seldom seen pythonaster, which is a fun name, pythonaster. And then the smaller one would be a goniasterid, possibly Bathyceramaster. So we collected them, so that should help with the final identification, as well as being able to run isotopes on both the sea stars and the sponges to identify if it's actually predating on the sponge versus just eating something else.
Kind of wonder when you've got several species of sea cucumbers here in decent numbers, kind of all in the same place. I gotta wonder what the niche specialization is during the different species, kind of how they,
It's a
what
cute
the
little
overlaps
sponge
are.
lollipop. What did you say? Oh.
You gotta stay long enough for school to get it.
Mm-hmm. Ooh, looks like a live sponge stock. Lots of sponges right now. Do you think that's a miniature version of the?
Does this remind you of karaoke night?
Yeah, all kinds of. Very spongy. There's a little sea star on the rock, like the one we saw on the that we collected. The coral that's eating the sponge. Not the one we just collected with the one before. Let's look down the center. Looks like there's somebody in there.
I can't even tell what it is.
No.
I see something.
Looks like sediment actually. I don't see the shrimp.
I don't think those two big things were.
Oh, those little tiny?
Oh, there's something deeper too.
Whoa! Oh yeah, you're right, there it is.
Found it.
That felt like going into another dimension.
Sorry if you got seasick.
I didn't.
You said you wanted the fast forward version.
I know. You're doing great. Little crinoid. Yep. Let me tilt this up a little bit. Another sponge. Dead stock. I wonder if like when if those like break off, you think they get like eaten by the sea stars on the ground? So I don't really see like dead sponge.
Well, we've seen several largish stalks that have been decaying, and some of the Phorea that's broken up.
But we don't see the bushel part.
That's true. I also wonder how much that just tumbles down with the current, maybe.
Yeah. Yeah. These Euplectellids are loving this space, though. They all look great. There's a little sponge bush. Do you want to look at that
Well,
sponge?
no, that coral is actually what I was pointing out.
I think that's what we just collected with the sea star
Okay.
on it.
Alright.
Yeah. A primnoid. Which we also collected before for the experiments.
You wanna look at this one?
No, we're good. That's enough.
Sorry, this is, so we need to give it a name.
Yeah.
Hang on.
Wanna look at this one or no?
It is, but let me explain.
Do we need to wait?
Don't need to.
Okay. No.
Yeah. With various nuts and bolts.
So. I wish I could do that.
So you're free to do some magnets on here. But you can just make those loop on from 4 to 3 potentially.
Just decapitated.
Yeah, now that you asked the question, now I'm really now I'm really curious.
There's a little sea star down
Mermaids using as didgeridoos.
there. Huh?
Mermaids using as didgeridoos.
Probably. I feel like.
We'll tell you the story later.
I'll put it in manual.
Oh, you sure?
Focus, yes.
Well, you're in ship. That's 3.7.
Can you brighten it up just a touch?
Oh, I kind of like it like that.
I did too, but the frame grabs look so dark. I'm not sure it's recording. It looks great on our monitor, but I think our monitor's running a little hot.
Need more?
No, I think that's fine.
That's okay. That's good. I think that's good for us to run. Oh, is it gonna boop us?
So this is a sea cucumber, holotherian, and these, I believe, are the type that spend a lot of their time swimming through the water column, land, ingest some sand,
This is an amazing
take
view.
back off, and then just float around digesting until they're ready for some more sand.
As you can see, the sand in its gut, since it's transparent, it's getting its daily exercise. That was a great shot, J-Rod.
Basalt with sponges.
Back to rocks. Venus flytrap. And another type of anemone or cup coral down there. Yeah, they do.
Yeah, they are definitely unhappy looking deep sea denizens.
It's a different type of anemone. Fish hiding under that rock.
More decapitated sponges. With a squat lobster.
Yeah, a little squat lobster on there. It's okay. I've really just seen some of these Norella, the little primnoids, but we haven't really seen any other coral since that Chrysogorgid where we sampled. Take a quick huh?
Little oaf on the
Yeah.
substrate below.
Very big disc
Yeah, it is a huge disc.
for a little dude. That trail of sediment. Should somebody release some weight before taking off?
Gotta dump the
Okay.
ballast.
Now that you do.
Fish! Tripod fish, I think. Yeah.
Not seem to be pleased about being.
But the giant alien robot chasing him, yeah, I could see that being mildly disconcerting.
Yeah, here
And that's
guy's
all the
gonna
swim he's
land.
got.
Whee, yeah. That was
And
pretty
touchdown.
graceful. Oh. All right, off we go. Can we scooch that so we can see the? Sorry, I know you're busy. Thanks.
Almost
Do,
Cool.
do all the things.
Yeah. Oh, there we go. Thanks, James. I always like when Sebastian's like embedded in the
There's a submarine.
another glass sponge. Looks like, yeah, I'll take a quick.
Talking like on the edge of the bathy in some cases, but I'm not sure what's going on here, but in terms of steepness,
Do you need me to come out?
there are no bridges that I'm not sure if we're in the bridge or not.
Oh.
It's just shallow or just but that's true. But in terms
I
of
think this is actually it's a primnoid.
so these, so this kind of wash area here, here,
No, I think that's okay.
they're gonna be smoother, and then you get up now.
Okay. And then we've got a bit of a wall here, or just a little pop up. There's a sea star on a rock.
The steepest part from this bathy seems to be from this bump to this bump. But I think any ridge you do there is fine. I worry about it getting really smooth and kind of like flat this area right here. But that's it.
This has been the spongiest dive.
It is. This section of the dive is very.
No, not there.
Lots of these euplectellid face sponges.
Second, 2/3.
And those little lollipop sponges, which I don't know the name of. Huh?
Not being in the Gulf of Mexico, I miss not having Bob Carney freaking out over the sea cucumbers.
Really?
Yeah.
It seems like there's like these fish that kind of like to lurk. Oh no, I'm just. We can look at this one. Just. He's probably not very happy about the lights.
I like a slight preference for this ridge, but for this ridge. But there's
Yep.
there's some like weird shadow bathymetry here. I'm not sure what's going on entirely. Yeah. So there's always sort of a question about that, but I mean,
Okay, that's good. He's gonna be pretty unhappy by the lights.
whole thing on the west side to the tank. It's just this one, Bobby.
Oh yeah. Quick zoom.
It's steep.
It's just kicking back. All right, that's good. That was a funny joke, guys. I always laugh at it.
Just kicking back. Sorry, I was trying to lay down. I heard it.
You're trying to work.
I heard it, I promise. What's that?
Yeah, it looks like dead sponge. That's a crinoid, yeah. Pretending to be a coral. just need to take one NIST scan when we're like 10 meters
Just
off the bottom.
let's fire them all.
Okay.
Or I guess how many are fired?
2.
I don't know if we need to fire
Yeah.
3 and 4.
We'll fire number 3 and number 4.
So everyone watching at home, we're gonna wrap in this dive here. Take one more water sample and start heading for the surface. And then we've got about a 11 or 12 hour transit and map after we get on the surface. So it'll probably be, I don't know, 12, 13, 14 hours till we're back in the water. About 70 miles south of here tomorrow.
And you just click on where you want to.
So have a good night, everyone.